the transgenic plants can be regenerated only from cells competent for transformation and regeneration. So if you have a plant which is not able to take up these genetic materials that you have been able to, inject into them, then it's going to be a huge challenge getting the plants to, or better so, the host cell to be able to express the characteristics that you have desired and you have introduced into it. You also have it that another bottleneck is that not all plant cells are competent for regeneration. So not all plants are totipotent in essence. So the third point is the relative population of the totipotent cells in tissue is determined by plant species and its genotype, type of organ, developmental stage of the organ, and growth history of the experimental plants. So before you do that, you have to consider all these before this introduction is actually done. The genotype, the type of organ that you are considering, through which you want to introduce, and the developmental stage of the organ as well. Perhaps you can look at the history, just as it has been stated, the previous history concerning the introduction of the genetic material into them. Now, generation protocols have to be a tailor-made for individual plant species. So it doesn't go, it's not like one jacketed thing that every plant would have to get, a protocol that every plant should get. You should get it for every species of plant. You should have a specific protocol that you're going to use to introduce such desired traits into them. So whatever method that you use for introduction of the desired gene into maize plant or maize plant tissue, it can't be the same for orange plant tissue. So you would have to, design a specific protocol for introduction of these genetic material to different species of plants that you are considering or you consider transforming. So that is for the bottlenecks. Now let's look at the plant tissue culture. Now, how do we culture plants? And when we say we are culturing plants, what exactly do we mean? So plant cells and tissues possesses some characteristics that enable them to be cultured in vitro in the laboratory. So when you say in vitro experiment, we do those things in test tube or in beakers. So that's in vitro. So cell possesses the full genetic potential of organism and under appropriate conditions can lead to the development of a whole plant known as totipotency. So any part of the plant, any part of the plants, all the organs, the roots, the stem, the leaves, okay, any parts, the flowers, the anthers, everything. All these parts of the plant, we expect it to carry some cell, totipotent cells that can regenerate into full grown plant. So with plant tissue cultures, small section of tissues, which is where you pick this, the sample from, we call it explant, are removed from an intact plant under sterile conditions and then placed on a sterile growth medium, leading to division and proliferation of cells present in the explant. Like I said, the explants are those plant materials that you want, some desired characteristics from, or you have extracted the desired characteristics from. They become our explants. Now, those ones that you have been using to be in the leaf, where you plug the leaf and all that, they become the explant that we are actually using for our experiments. So let's look at, briefly, what this transformation is all about. So you, from your explant, which is, in a potted form, you have your tissue, excised from it, we cut your tissue from it or your cells from it, and you have your medium This is the medium, I mean the blue tray or the petri dish. This is what we see at the lab, the petri dish, or that's what we use at the lab. You have your petri dish containing your medium, okay, your preparations through which the plant material will, the plant tissues will form calluses. Now, calluses are undifferentiated cells. Calluses are the undifferentiated cells. But in this medium, what you have to do is that after callus formation, after the cell has been undifferentiated, but you have a grown mass of cells, you apply some plant hormone or plant growth factors. Example is auxin. So when you introduce auxin, what is going to happen is that you foster, or better so you facilitate, a root and shoot elongations, okay, or root, shoot and root system formation. And so you have your plantlets. So out of numerous cells or tissues that you have in the, I mean, petri dish, I mean, forming calluses, you have your plantlets grown out of it, and you can have many adult plants grown out of the very small plant tissues that you excise from the explants. Okay. Now let's look at types of plant tissue culture. Types of plant tissue culture. Now, different types of plant cultures can be generated based on the initial type of plant tissue used for the explant and the composition of growth medium. So the composition of the growth medium one influence the sort of culture that you are going to, I mean, embark on, and then also the type of explant or the form of explant that you are going to use. Whichever organ that you are going to use from the explant is also going to influence the types or the type of culture that you can adopt. One, you can have callus culture, you can have cell suspension culture, you can have protoplast isolation and culture, you can have anther or pollen culture, and you can also culture the plant organ. You can also culture the plant organ, being the root, being the stem, being the leaf. You can also culture them. Well, we are going to look at them one by one. That is the types of cultures that we have. So number one is the callus culture. That is what we are going to, I mean, look at. And I have told you that callus culture, with callus culture, we are looking at undifferentiated mass of cells, okay, which are yet to be differentiated into various plants, I mean, plants forms, or better so, the plant parts, the roots, the stem, and also the leaves on the branches, fruits, and all that. So aside that, before that begins, you have it as a callus tissue, or the undifferentiated form of cell or mass of cell yet to be differentiated when a plant's hormone or plant growth factor is introduced into the medium. So let's look at the plant culture now. So an explant usually containing a region of meristematic cells. Now, meristematic layer of the plant is where continuously and consistently cells are divided, and later they get to the region of elongation, they get to the region of maturation, and also the differentiation. So it is incubated on a growth medium containing certain plant growth factors, such as the auxin and cytokinin. And I have just mentioned the auxin. The auxins, I said, is responsible for shoot and root elongation. So the cytokinin is also responsible for cell division. They are all plant hormones that we introduce into the growth medium, or better still, the, I mean, callus culture medium. The cells grow and divide to form an undifferentiated mass of cells, and this is called, like I said, callus. Right. If the cells are periodically transferred to the fresh media, you have it differentiated into forming the plant organs like the roots, the stem, and the leaves. So if you have a question, I would advise that you don't type in the message box. You raise up your hand and you ask the question for clarification. You raise up your hand, kindly raise up your hand if you have a question, and it will be duly addressed. You'll be called upon and will address it accordingly. Thank you very much. Right. So the cells can be directed to form or differentiate into roots or shoots, and the process is called the organogenesis. And later, it is transferred to a growth medium containing different combination of plant growth regulators. These plant growth regulators, we also refer to them as growth factors. And in some, articles, books, and all that, they may also refer to it as plant hormones. So whether growth factors, growth regulators, or plant hormones, we are referring to the same thing. Now, these hormones, we have the cytokinin, we have the auxins, we have the gibberellins, and ethylene, and the rest. They all have their specific role as far as the plant growth is concerned. Right. Please, if you have any question, don't hesitate to, as I go on, don't hesitate to just put your hand up. I'll call you and I'll clarify if there is any concern. Okay, so we are moving on. So these are, simple process that we can culture plant tissues. So you can have, as an example, you can have the carrots, and we can cut the carrots into some small pieces, and you have it incubated, or better still, put in a prepared medium in a beaker. Now the beaker has your medium, and this medium contains, number of combinations, number of factors. You have the plant growth factors, you have the the carbohydrates, you can have some antibiotics in there, number of things are in there. These antibiotics will inhibit the growth of microbes, and then also the carbohydrate source is serving as energy for the tissue or the plant part that is placed in the medium, because it will need it for respiratory activities. And then also we have many other composition as well. We'll look at the composition briefly. So in subsequent, because you have the very tissue that was, kept in the medium, developing some culture, and at the end of the day, finally, when you look at the final diagram to your far right down, you have some portion of some tissue forming or differentiating to form the roots, the stem, and also the shoot system, the stem and the leaves, which are the shoot system. So that's what actually we have for the culture. Then let's look at the suspension culture. We have learned about the callus culture. The callus culture, you are not actually interested in forming the various plant parts. That is the roots, the stem, and then the leaves. You are just interested in the mass, the bunch of undifferentiated mass of cells increasing and dividing continuously to give you that mass of undifferentiated cells. But then if you have the cell suspension culture, what do we have for this one? We have made mention that you have different types of plant tissue culture. So plant cells are grown as individual cells or small group of cells in a liquid culture medium. This is initiated by transfer of plant callus into a liquid medium containing combination of plant growth regulators and chemicals that promote disaggregation of the cells into single cells or small clumps of cells. Now, continued cell growth requires that the liquid culture are shaken at low speed to promote aeration and chemical exchange with the medium. Plant cell cultures can be used to produce whole plants through a process called somatic cell embryogenesis. Now, this is the cell suspension culture diagram that we can have. Okay, so you have these flask, and with this flask you have the callus culture placed in the medium. Each of these flasks is having prepared medium. So you just pick your callus culture and the sterile condition, whatever is done. Actually, in the lab, we work in the flow hood, and in the flow hood, you make sure that the whole place is sanitized, the whole place is kept so clean, and outside air is, as a matter of fact, being avoided. That's why we are working in the flow hood, so that the flow hood will pick up the outside air, sanitizes or disinfect it, and aerate the medium with the air which has been cleansed, or better still, sanitized. We also have the protoplast isolation and culture. The protoplast isolation and culture. With the protoplast isolation and culture, we have the protoplast of plant cells whose cell walls have been removed, usually through enzymatic processes. So you can have some enzymes, lysine enzymes, to remove the cell wall of the plants, so that the protoplasm, or better the protoplast, will remain without any cell wall. So protoplasts isolated from different plants are forced to fuse together to form a hybrid. The hybrids formed from protoplast fusion can represent genetic combination if they are generated into whole. Yeah. All right. So we have the two. We have two different genetic combinations, or genetic combination of two different traits, yeah, being fused. And at the end of the day, you are going to get the hybrid of the two. So protoplast fusion provide additional means of genetic engineering, allowing beneficial traits from one plant to be incorporated into another plant. And in that sense, you would have different, traits being exhibited. You have this diagram on your screen. So you have the protoplasm, or the protoplast of different plants down here. And these are cultured in a medium and are fused. You realize that you get one plant, or better still, one cell which is having combination of genetic material from different sources or different cells. And at the end of the day, when they are cultured in a medium, you have a callus formed, and when growth factors like the ozone, cytokinin, and the rest are introduced in the medium, differentiation of these cells into plant parts will start forming, and then you get your roots, your stem, and other shoot system, and you get your plantlets growing, and then you can transfer them into uh, your fields when they are well grown. And then you have another one called the anther or pollen culture. In this culture, anthers are excised. When we say excised, we mean they are cut or they are extracted from flowers of plants and then transferred to appropriate growth medium. After a period of time, pollen cells are manipulated to form individual plantlets. And you know that pollen is it's not diploid, pollen is haploid. So plants produced from anther or pollen culture may be haploid because they were originally derived from pollen cells which have undergone meiosis. And when we say haploid, we mean that half of the genetic composition of the organism. So in human, we have our haploid cells to have 23 chromosomes, not 46. So such plants may be sterile and not useful for genetic manipulation, but they can be treated at early stage with chemicals that allow chromosome duplication. And this is the process that we can, use to undergo or undertake the pollen culture. So from the up there, you have A, and then that's the anther. Then you cut it. At the end of the day, you excise it and you introduce it in a medium. That is the F down, and then it will start regenerating into the plant parts or differentiating into the plant parts. And at some point, you have to just transplant into a stable medium and supply it with some nutrients, and it will start growing into a full-grown plant. Then you also have the plant organ culture. The plant organ culture is the growing of plant organs under culture conditions. For example, section of plant root can be excised and transferred into a liquid growth medium where the roots will proliferate excessively to form both primary and secondary root branches. And I'm sure some of you have come across bryophyllum, and we used to perform some experiment with bryophyllum. We keep it under a very good condition, and roots or rootlets will start forming from even the root, the leaf of the plants that has been kept in some appropriate medium. And we have worked with bryophyllum. I'm sure most of you have also worked with bryophyllum before. Yeah. So that's what we have on our screen. So you have the plant organ in the A up here, and then you have excise the plant parts or the plant organ. It can be the root, it can be the portion of the stem, and also the leaf. And then you incubate it, or better so you place it in a medium, and callus will be formed. The callus will be formed, and you can now transfer the callus into a medium that you can harness it to become a plantlet and subsequently become a full-grown plant. So plant tissue culture media, and this is where we get a combination of factors that promotes the growth of the plants in that particular media. Okay. Charlotte.
Hello, Doc. Yes,
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 Charlotte,
Doc.
I see your hand up.
Yes, thank you very much. Please, where do we classify budding and grafting?
So, budding and grafting, we are talking about a vegetative, um, part of propagation. It goes beyond. When we started this particular course, I told you that we are not looking at the vegetative form of propagation, like you get the sucker, you get the stem cutting, you get the bulb or something, and you want to propagate it. It goes beyond that. Biotechnology goes beyond this. Biotechnology, we are looking at combination of genetic characteristics. Okay, a desired gene that you have observed in a different organism, and you want a plant of or a host plant to also exhibit that characteristics. And I give example, and some of you even give an example. This orange melon. Now, if you have your orange and also your lemon, and then you do budding, you cut some portion of the lemon, and you bud it with the orange, and at the end of the day you have the orange growing on the lemon plants. That is not, that is, we don't consider it as some form of biotechnology, because no genetic this thing has been introduced. Okay, it's just a support. The lemon is just a support. The lemon plant is just a support for maybe uh water or something, provision of water for the new plant that is growing on it. But no genetic cross or better cell genetic combination has been done. But with biotechnology, we are looking at infusing a genetic material into a different plant. Charlotte, do you understand what I'm saying?
Yes, Doc.
We're looking
Yes,
at
Doc.
infusing a genetic material, a whole genetic material into a host plant to exhibit certain characteristics. So when you
Okay.
bud, that is not, we don't consider it some form of biotechnology. Uh, then when you also introduce, or better so, you have a bulb of onion and you propagate it, it's the same genetic characteristics that you are going to get from the offspring. If you have cassava and you cut the stem and you propagate it, you're still going to get the same thing. But the situation where you consider the cassava, maybe the growth rate, or better, the maturity or maturation period of the cassava, and you go and introduce maybe a gene which is able to shorten the maturation period of that cassava, that is when we say that you are, doing genetic engineering, or you are engineering the cassava crop. And that is where biotechnology actually comes in. Is that okay?
Yes,
That
Doc.
is where
Thank you very much.
biotechnology actually comes in. Right. Theophilos, is it Theophilos or something like that? Is that name correct? Can you pronounce
Tianistus.
your name for me?
The name is Tianistus.
Tenistos.
Tianistus. Please, it's my daughter's laptop that I'm using. I'm Juliana Egan.
Right. Okay.
So I want to ask a question.
Okay, go ahead.
Uh, Concerning this biotechnology, where do
Great.
we categorize the production of this mushroom that we buy from the market?
Mm-hmm.
This new mushroom that has been introduced.
Okay.
Which type of culture is that?
Which part of what?
This culture we've looked at the colors culture, these different
Plant
types of
organ culture, the protoplast
culture.
culture, and the rest.
So where do we categorize that type of production? Does it
Very
fall
good.
into
So we
this?
are looking at, yeah, in the first place, mushroom is not a plant. Mushroom actually is not a plant, it's a fungi. Okay.
Yes, please.
It's a fungi, it's not plant. We are looking at plant tissue culture specifically. Mushroom is not actually a plant. It's a fungi. So it doesn't
Thank
fall under any of these culture that we are discussing right now.
Yes, please. Thank you very much.
Great, great. Right. So let's look at the media. We have been mentioning media, and then you prepare the media and you place it in the, beaker or in a petri dish. Now, how do we prepare this media, and what is actually the composition of this media to support this type of culture that we have actually discussed above? Now, the culture media are largely responsible for the in vitro growth. And I have explained the in vitro growth. I mean, the growth that occur in test tubes, in beakers, and also in flask, whether round bottom, bottom, and all that. They are the in vitro form of propagation, and also the morphogenesis of plant tissues. Now, basically, the plant tissue culture media should contain some or same nutrients as required by the whole plant. And it may be noted that plants in nature can synthesize their own food material, of course, through the process of photosynthesis, which we all know. But however, plants growing in vitro cannot photosynthesize or cannot prepare their own food, right? Because they don't have these green pigments like the callus. The callus is not having this green pigments for this photosynthetic activity to occur. So what you have to do is that you have to supply the medium with some plant nutrients. And that is what we are actually going to look at. So let's look at the composition of media. So the composition of the media is primarily dependent on two parameters. And what are the parameters? The parameters, we have one, the particular species of plant that you are considering. And number two, the type of material used for the culture, whether the tissue, whether the organ, or whether the protoplast that we have discussed already. If you are also using the callus culture, the media is also dependent on what type of culture that you want to embark on. And thus, the composition of the media is formulated considering the specific requirements of given culture system. The media used may be solid. It can also be a liquid media. Now, let's look at the type of media and a type of culture that will favor such a media. So, white medium, we also have the moisturage and scotch medium. We have different types of medium which we are going to look at. So let's look at the white medium. And this is one of the earliest plant tissue culture media developed for the root culture. In essence, we can also say the organ culture or the plant organ culture. We use the white medium. Now, we also have the MS medium. All these preparation can be done at the lab. They sell the composition or the formulation. So you can just walk into maybe an industry, or better still, a workshop outlets, and then get these compositions or formulations. And you can come back to the lab and prepare these media yourself. So the Mushij and then Skug originally formulated a medium to induce organogenesis, that is organ formation, the tissue, the roots, and then the shoot systems, okay, and regeneration of plants in cultured tissue. So, but most of the times, if you come to the lab, this is the medium that we mostly use. The MS medium is what we mostly use for organogenesis, as formation of the organs of the plant. We also have the BF5 medium or the B5 medium, and it was originally designed for cell suspension and callus cultures. At present, with certain modification, this medium is used for protoplast culture. And we have the NSIS medium. You also have the NSYS medium. Matthew, I can see your hand up. Is there any concern? Matthew, your hand was up. Matthew, your hand was up. Hello, Matthew. Can you hear me? Can I continue, Matthew? Okay. Can you hear me?
Sir, please continue. Maybe his network.
All right. Okay. Right. So let's look at the NSYS medium. So this medium is used for cereal anticulture besides other tissue cultures. So you realize that this medium that we have been talking about, the media that we have been talking about, they are for specific plant tissue culture types. And we also have the niche medium, and this is frequently used for anticultures. So the NSYS medium, the niche medium, and also the other medium, we have them used for the anticultures. But generally, the MS medium is used for, you can use MS medium also for anticulture as well, and it will work out perfectly. So that's it. Let's look at the constituents of media, the composition of the media. Now, the composition, we have the inorganic nutrient that is going to be required if you want to prepare the media. The inorganic nutrient consists of the macronutrients and the micronutrients. And the macronutrients, or the elements, we have the nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, and some essential micronutrients for the tissues of plants. Right, so I've mentioned some of the macronutrients that are going to be very beneficial as far as the plant tissue culture is concerned. Let's look at some of the micronutrients as well. Now, the micronutrients which will be needed, you have the iron, and these are actually needed in small quantities. So iron, manganese or manganese, zinc, boron, copper, and molybdenum, they are also very crucial or critical in plant development. And the chelated form of iron and copper are commonly used in the medium. Now, you also have the organic medium. Carbon and energy sources are all the organic medium. We've looked at the inorganic media or media nutrients. Let's look at the organic medium nutrients or media nutrients. So plant cells and tissues in culture medium are heterotrophic and therefore are dependent on external carbon for energy. Now, if they start producing their own food, we refer to them as autotrophic because they prepare their own food. But once they depend on other organisms for food, then we refer to them as heterotrophic. Among the energy sources, sucrose is the most preferred. Sucrose is the most preferred. Right. Organic supplements. The organic supplements include vitamins, amino acids, organic extracts, activated charcoal, and antibiotics. They are all important ingredients that is going to be used as far as the formulation of the plant culture medium is concerned. Can I have Philomena muting herself? Kindly alert Philomena that we are getting feedback from her end. Now, let's look at the plant growth regulators. I also said that the plant growth regulators, if you don't want to refer to them as the plant growth regulators, you have them as the plant hormones. They can also be referred to as the plant hormones. Right. The four broad classes of plant growth regulators or hormones used for the culture of plant cells are: we have the auxins, we have the cytokinins, we have the gibberellins, we have the abscisic acid. We also have the ethylene, but actually it is actually needed when you have the fruit ripening. It's in a gaseous state. So when banana is ripening, these are exuded. This plant hormone is exuded in the gaseous form, and it causes ripening of other fruits as well, or other bananas as well when they start ripening. So auxins, very, very crucial. So at a low concentration, auxin promotes root formation, whilst at higher concentration, callus formation occurs. So if you want to undergo or embark on callus formation or callus culture, concentration of auxin should be in high concentration. All right, cytokinin, they are involved in cell division and shoot differentiation and somatic embryo formation. And what does the gibberellin also do? They usually inhibit adventitious roots and shoot formation, and they induce dwarf plantlet to elongate. And then the last but not the least is the abscisic acid. To enhance shoot or bud proliferation, abscisic acid is actually required. Right, let's look at a solidifying agent if we are working on the solid medium. Remember, the media, we have the liquid medium and we have the solid medium. So if we are working on the solid medium, after all these ingredients, we need something which will solidify it for us. And we use the agar, or the polysaccharide obtained from the seaweeds, and they are used as a gelling agent. And then we also have to consider the pH, the alkalinity, or the acidic medium of the media. Now, there are some crops that grow in basic medium. There are some crops that also grow in acidic medium. So you have to also consider by measuring the pH of the media. The optimal pH for most tissue culture is in the range of 5.0 to 6.0. At a pH higher than 7.0 and lower than 4.5, the plant cells stop growing in cultures. That is to say that the media shouldn't be too acidic or too alkaline. If the pH falls during the plant tissue culture, fresh medium has to be prepared. So essentially, this is what we have for the plant tissue culture. And if there is any concern, I will share with you. If there is no concern with regards to the plant tissue culture, we'll just then move to animal tissue culture, how to culture some tissues in animals. Prosper, do you have something for us? Prosper, do you have something for us? All right. I think we have to move on to the animal tissue culture. All right. The animal tissue culture, unlike that of plant, is going to differ in a bit. So many animal cells can, with special care, be induced to grow outside their organ or tissue of origin. Isolated cells tissues or organs can be grown in vitro when they are kept at defined temperatures using incubator and supplemented with medium containing cell nutrients and growth factors. Now, cell types cultured include connective tissues, and these connective tissues include bones, cartilages, blood cells, blood, and muscle cell, epithelial cell, neural cells, and many type or different types of tumor cells. Now, we also would want to consider some tumor cells. Not all of them are, cancerous. We use some of these tumor cells for, scientific researches, like the crown gall in plants. Okay, so let's look at types of animal tissue culture. Animal tissue culture is used as a generic term to include the in vitro cultivation of organs, tissues, and cells. So it doesn't mean that when we say tissue culture, then strictly we are talking about only the tissue, cultivation or incubation of tissues only. But it is a general term that is used to refer, cultivation of either organ, tissue, or cells. Now, animal tissue culture can be subdivided into three major categories. We can have the organ culture, we can have the explant culture, and cell culture. Now we can go on and look at the organ culture. The whole organ or part of the organ is maintained in a way that allows differentiation and preservation architecture. So the animal tissue actually does not change. It is actually maintained when it comes to the organ tissue culture in shape and in exact form. Now, the basic equipment and facilities that are needed would be discussed later. These tools will facilitate and enable the cultivation of these tissue, organ, and cells. Now let's look at another type apart from the organ culture, which is the organotypic culture or the explant culture. Now, in explant culture, small pieces of the tissue of interest are simply allowed to attach to an appropriate substrate, usually one that has been coated with collagen, and collagen is a protein, and in a rich medium of serum. Now, when we mention serum, we are referring to the medium that doesn't contain, cells. It has all the protein compositions, but without cell. So, cell culture, we can now go on and look at the cell culture. The cell culture or the cultures are divided from dissociated cells taken from the original tissues. Cells are dispersed mechanically or enzymatically into a cell suspension, which may be then cultured on a solid substrate. So like I said, there are some equipment and basic facilities that we need to work and to ensure that we don't introduce some pathogens or some microbes into our environments which we are working. Now we need a sterile working area equipped with airflow cabinets and a lab coat. And in this, we can use the flow hood. So we also need the incubation facilities to maintain the cells at a required temperatures, and refrigerators and freezers, which is below 20 degrees Celsius. Both items are important for storage of buffers, liquid media, enzymes and some media components are very sensitive to high temperatures. So what we have to do is to keep them at very, very low temperatures. You know, enzymes denature when the temperatures are very high. They are proteins. We also need microscopes, and it is a vital instrument to be able to recognize morphological changes in cultures to assess progress or otherwise of those cultures. Now, tissue culture wares that are going to be required include plastic glasswares, and they should be used in support of the cell growth adequately and must always be chosen to match the procedure. Then we also need the sterilizing facilities like the glasswares, and they should be soaked in a suitable detergent. Right. Now, liquid nitrogen, or we can also have a replacement as the defreezer. Samples of cultures will need to be frozen for storage to prevent genetic drifts and to guard against loss of the cell line. Now, we also need distilled water or deionized water. The pH of the double distilled water should be regularly checked, as in some cases, this can vary. Now, facilities for counting the cells should also be provided. And the device which is used in this, cell counting is the hemocytometer. And this is designed for counting blood cells. Now, we also have the general items like the water bath, centrifuge, vacuum pump, shaker, graduated pipettes, and many other items that are going to be beneficial as far as the tissue culture or animal tissue culture is concerned. So all these are very important. Now we look at the culture media. Now, basic culture media, the most basic media are balanced salt solutions, which is the phosphate buffered saline, popularly known as the B or the PBS. And they are used for washing cells and for short incubation in suspensions. Now, we also have the buffering capacity, and cell cultures have the optimum pH of 7.4 to 7.7 for the general growth. So the type of buffering that is used for the media depends on growth conditions. You also have the serum, like I mentioned. Most cell lines still require serum for adequate growth. Various sources of serum may be used, such as the calf, fetal calf, and horse. The level of serum used depends on a particular cell line. And as well, you need some antibiotics in the media. Unlike good sterile condition can be maintained, it is necessary to also incorporate antibiotics into the medium, so to prohibit outgrowth of some bacteria. You also need some amino acids, which include glutamine and other forms of amino acids. Commonly, the necessary amino acids include the cysteine, we have the tyrosine, but some non-essential amino acids may be needed. Glutamine is also required by most cell lines as energy and carbon source. And it's all right, so let's look at some sources of animal tissue culture. So we can get it from adult tissue. We can get these tissues also from embryonic stem cells. We can also get it from normal tissue. I mean, example is the fibrocytes or the endothelium. Now, we can also get it from the neoplastic tissue, and these are the tumor tissue that often provide cheap and simple method of producing large number of cells. Now, let's look at uses of animal tissue culture. Why do we have to even embark on culturing animal tissues? Is it to produce new human or to produce new organism like a dog or cat or ant or elephant? Now, we culture this tissue of animals and they are used as model systems. We also use to study bacteria, viruses. And so, as a model system, cell culture are used to study basic cell biology and biochemistry, to study the interaction between cell and disease-causing agents like bacteria, virus, and also to study the effect of drugs, that is toxicology. Okay, and then as well as pharmacology. And then also the process of aging, and also it is used to trigger, the stage of aging. Then with the virology, cell culture can also be used to detect and isolate viruses, and also to study growth and development cycle of viruses and their mode of infection in humans and other organisms as well. Now, for, like I said, or as a model system or as a model organ, apart from its use as a model organ, we can also look at the toxicity testing of some drugs on these cells. So mammalian cell cultures are used to establish the potential toxicity of compounds and also to study maximum permissible dosage of new drugs, the effects of new drugs, cosmetics, chemicals on survival and growth of number of types of cells, especially liver and kidney, which play very important role or key role in excretion or eliminating toxic substances that are accumulated in our body tissues. Then we also look at it in this sense as it helps us to produce vaccines for human and other organisms. Cultured animal cells are used in production of viruses, and these viruses are used to produce vaccines. For example, vaccine for deadly disease like polio, rabies, chickenpox, measles, hepatitis, the corona or the SARS, and then also other viral infections or disease are produced using animal cells. So before any drug is produced, they use these cultured cells to test them and then as well check their toxicity level before they are administered to a live organism. Now we also use them as, in genetical engineering. So animal cell cultures are used to produce commercially important genetically engineered proteins, such as the monoclonal antibodies, insulin, hormones, and others. We also culture these tissues to replace tissue or organs. So animal tissue culture can be used as replacement tissue or organs. For example, artificial skin can be produced using this technique to treat patients with burns and ulcers from wounds. It can also be used for genetic counseling. We culture animal tissues and we use it in genetic counseling. So fetal cell cultures extracted from pregnant women can be used to study or examine the abnormalities of chromosomes. And these findings can be used in early detection of fetal disorders. We can also look at gene therapy, but this is quite expensive. So cultured animal cells can be genetically altered and can be used in gene therapy techniques. Now, gene therapy involves altering DNA within cells in an organism to treat or cure diseases. So let's be hopeful that in the near future, sickle cell issues will be resolved through this technology, the gene therapy technology. And that brings us to the end of our unit 2. Is there any question or concern or contribution that you would want us to highlight on? Is there any question? Can we move on?
Yes, sir.
Yes, Doc.
Yes, sir, we can continue.
I think
Yes, Doc, please, we can
Delia,
continue.
your hand is raised.
Yes, sir. Sir, thank you.
Samuel, your hand is raised. Okay,
Sir,
Fidelia, let me listen to you.
sir, please, when I joined, I went off again, but when I came back, you were talking about this culture issue. And please, I want you to just give me an explanation of it, what it means, the culture.
Culture is simply cultivation of tissues or organs or cells by storing them under sterilized conditions. Okay?
All right, sir. Okay.
And then when you keep
Okay.
them under these sterilized conditions, what you have to do is that because they are cells and they are living things, you have to supply them with nutrients. Now, we have mentioned some of the nutrients. Some of them are organic nutrients, some of them are inorganic nutrients. You also have to supply them with energy sources, like the plant tissue when you culture them in vitro, okay? Or when you are culturing them in, I mean, test tubes, when you are culturing them in beakers or flask, what you have to supply them is energy source like the sucrose, which is the most preferred one, so that they can continuously grow. You don't culture them for them to die. You culture them for them to still be alive so that you can study them, as in the case of animal tissues. Fidelia, do you get it?
Yes, sir. Yes, sir.
Great,
Okay.
great.
I'll follow this. Thank you.
Okay, right. Can I have Samuel? Samuel,
Yeah, yes, sir. Yes, sir. Sir, please, I want to ask that since the animal culture includes microorganisms like
come
bacteria,
again. Come again.
I'm asking that animal, when we are starting with the animal culture, we have microorganisms like bacteria, fungi, and the rest. I'm asking that
Okay.
can it contaminated, and if it can be contaminated, how can we prevent the contamination?
Yes, I think we spoke about that. That to prevent the contamination, what you have to do is that you have to introduce antibiotics. Aside you working in a sterilized environment, you still have to use antibiotics to prevent growth of unwanted microbes. Okay. Hello, Sammy.
I can hear you, sir.
Okay, did you get what I said, please?
Yes, sir. Yes, sir.
Great. So you introduce antibiotics into your medium that you have prepared, whether the NSIS medium, whether the MS medium, whether the B5 medium, or whatever medium that you are going to use based on the type of culture that you are embarking on. You have to introduce specific antibiotics so that it prevents growth or outgrowth of other microbes as well, which are not wanted or which are not needed in your experiments. Right. I can still see my Matthew's hand up. Matthew, is your hand up? Okay.
So maybe
Right.
it's a legacy hand. He's not aware.
Okay. Right. So I think we can continue. Please, can we continue?
There is a hand up.
Yes, Doctor.
Yes, okay.
There is a hand up. Okay,
Yes.
Emmanuel, Katie. Emmanuel.
Yeah, yes, sir. So mine would be a suggestion. I would suggest that maybe if at the end of a section or a unit, you can provide us with some maybe sample questions so that we can try to guide.
Oh, that's interesting. So indirectly, you are telling me to give you your end of semester exam to try your hands on. Is that what you are saying?
No, sir. No, sir.
No,
That's not so.
Trial questions.
I want
Not
to
the exam question, but
Trial questions.
So why can't you yourself, maybe in your groups, set some questions and try your hands on, but you want the questions to come from me? I don't understand.
Hello, Doc.
You want to
that's real question. That's real question.
Hello.
Yes, I can hear all of you. So you can talk. I can hear all of you.
So your father who your hello sir. Your
Yes,
father,
boss. I
Mr.
can hear all
Boaje.
of you. So you talk.
Yes. Mr. Boaje, who happens to be your father. He always give us sections for quiz, section for exams. And then he give you trial questions, tutorial questions that are similar in nature to your quiz and your end of semester exams. That is why my colleague Ima is just asking for not set questions that is going to come in the exams, but at least, just as Dr. Aboye has been doing, you give us, let's say, chapter 1 chapter 1 to chapter 2 is for quiz, chapter 3
Okay.
to the end is for end of semester, and then chapter 7 to chapter 10 is for quiz 2, and then he will give you sample questions to try your hands on. That is all that we are asking for. This is
Very
simple
good.
as ABCD, like man you win. Simple.
That,
Don't bring your mind with things here.
we are asking so that we will get some gist of as to how the questions will be.
Okay, but I think
If you're lucky, then some will come, and then if there's some
in any case, ever since I started teaching application of biotechnology, do you know that nobody has asked this question before? Apart
Because
from
we are
you.
special.
Yes, so that is how
Yes,
we
we are
are.
special class.
because we are special.
Okay, so why? So I'm sure you have come across some of my sample questions or end of semester, and then
No,
Oh,
No,
no,
not at all.
no,
not
So
at
we don't
all.
get that
but if
question.
there are
So
some, then please we have
Not
those so that
at
we
all.
don't worry you, Crapo.
okay, so okay, can you do me a favor? Set your own questions and forward them to me. answer them and I will look at them for you. What about that one too?
No, time factor. The time is not sufficient for us, so please help us.
You don't want questions, eh? You Fidelia, you don't want questions. Okay.
That's what I'm saying. Time factor. We can't set the question, so Sel should give us some questions.
Which one is the time factor? Which one? I don't understand. When you say time factor, I don't understand.
So we're sitting down and then setting the questions, please.
No. So
That
the time factor
is why.
Say the time factor.
That is why I have given you. That is why I have given you the slides. Nothing will come out of my slides. Everything will come from my slides that I have given you.
It's not easy. Voluminous.
The
truth of the matter is that it's not easy. We don't have time.
Can we
So the
continue?
tutorial questions help us more.
Looking at the scope of what we are learning, and then the
Anyway,
time.
let's continue with the lecture.
That is why we are saying is time factor. We are learning so much at the shortest possible time, and so that
okay, I'll see what I can do.
Thank you.
Yeah. Thank you, dog.
I can see what I can. I can see what I can do. Okay. You also, and then may you give birth to twins.
Amen.
No,
Amen.
I
vacated.
I beg, I'm close.
Okay.
Now.
I think we can now continue after enjoying. I think we can now continue.
So let's continue.
So we look at the chapter 3, which is application of biotechnology 1. And I have given out the slides already. So you can follow on your class rep or coordinator for your chapter 3. So let's look at the unit 3. The application of biotechnology 1. In this unit, we are going to consider application of modern biotechnology in agriculture, and then also modern biotechnology in industrial food processes. We will look at some fermented cereal foods, fish, and beverages in Ghana. And then we look at other fermented beverages and foods. We look at application of modern biotechnology in medicine and healthcare. And then we will look at bioremediation to end the unit 3. All right, so let's start with the unit 3. So application of modern biotechnology in agriculture. One reason is to enhance crop protection. One reason for application of biotechnology, that is the modern one in agriculture, is to enhance crop protection. So crops such as corn, cotton, and potato, and of course many other more, have been successfully transformed through genetic engineering to make a protein that kills certain insects when they feed on the plants. And the protein is from the soil bacteria, Bacillus thuringiensis. That's why most of the times we call these BT. So BT is not the same as biotechnology. BT is a short form of the bacteria which we get the protein, which is the Bacillus thuringiensis. So researchers from the University of Hawaii and Cornell University, for example, have developed 2 varieties of papaya resistant to papaya ring spot virus by transferring one of the virus's gene to papaya to create resistance in the plants. Now, we also look at the second one, which is improving nutritive value of the crop. Now, rice breeders are using biotechnology to build vitamin A. Now, you know vitamin A, is responsible for, preventing night blindness. And they have built this into rice, the vitamin A into rice. And this has improved the ability to produce beta carotene and is a precursor of vitamin A. Genetically enhanced soya bean that are lower in saturated fats and higher in oleic acid and more stable during frying without further processing have been also developed. So biotechnology is being used to develop potato varieties with a higher starch content than the normal one that we have been enjoying every day. We also have advantage of biotechnology in modern agriculture as to enhancing or improving harvests and post-harvest characteristics. So transgenic tomatoes allow farmers extra time to harvest and deliver tomatoes to the marketplace by interfering with the polygalactoseries. Now, polygalactoseries is responsible for pectin decomposition, and pectin is actually a polysaccharide. And when this pectin is decomposed, that is where we get a tomato getting spoiled. So when we are able to interfere with the production of this particular hormone, or better still, this enzyme, we are able to keep the pectin from being decomposed, and the tomato will be preserved for a longer period of time against post-harvest losses. Now, genetic modification has been used to slow down softening in other fruits such as apple, also in watermelon, thereby increasing shelf life during shipment and storage. Now, we have another advantage, or better still, importance of biotechnology in agriculture. Now, we also are able to let crops be tolerant to environmental stress. And when we mention environmental stress, we are looking at droughts, we are looking at salinity, we are looking at alkalinity, we are looking at acidity, and so forth and so on. High temperatures and all that. So, tomato has been engineered by Canadian scientists to grow in water nearly half as salty as the ocean. The modified tomato contains a gene that makes it gather anions inside large cells, and they cannot harm the plants. It's Charlotte. Your hand is up.
Yes, please. I want to find out, like for instance, this GM, the modified plants, like boiling down to the tomatoes. Some years, about two, was it? I bought some tomatoes, and then I noticed that when I stored, I didn't put them in the refrigerator. But the tomatoes were able to last for about 3 weeks without spoiling. What I observed was that it was rather shrinking, and that was a COVID era where you buy and then. So I think coincidentally I bought this, and it was not getting. It was not spoiling at all. So I saw it to be a very good. So I decided to extract the seeds and then plant. But I noticed that when I extracted the seeds, it didn't germinate at all. It didn't germinate. So I don't know whether sometimes these modified foods inhibit. There's a form of some kind of inhibition when it comes to having a viable seed for propagation. I want to find out, does it have the effect on that?
Yes, Charlotte, you are right. So as we just discussed, they interfere with the breaking down of the pectin. Okay, they inhibit the production of the enzyme which is responsible for breaking down of the pectin, which is the outer covering. Okay, now what happens is that your question is that why is that you were not able to propagate it with the seed?
Yes.
Is that your question essentially?
Yes, please. Yes,
Yes.
please.
So we will get to patency. You take your time when we get to the patency with regards to these GMOs, then we can discuss it in detail. Okay.
Okay, sir. Thank you.
Good. And surprisingly, that is going to be your presentation. Are you happy about that?
Hey.
Let's move on and see. Yeah.
Right. So that has to do with patency. They have to get some sort of rights over what they do as well. Okay, for economic purposes. For economic purposes. So it is not out of place that you wanted to propagate and it wasn't viable. It wasn't out of place at all. But we will discuss them in detail in the chapter 4 and 5. Sorry, the chapter 5 and 6. We'll look at it in detail. Right. All right.
Okay,
So,
yeah.
Charlotte,
Thank
if
you.
that's okay, we can move on.
Oh, thank you very much, sir.
Sure. Right. So, we also have better taste and flavor. That's the modern biotechnology in agriculture to enhance better taste and also flavor. So, taste and flavor can be altered by enhancing the activity of plant enzymes that transform aroma precursors into flavoring compounds. Now, transgenic peppers and melons with improved flavors have been on the field for trials. So, very soon, we are going to get those ones as well. So, increase farm yield. This one, it has been very long. I mean, crops, cassava, yam, potato, and those crops, GMO crops, they've been out for a very long time. So we have these ones already. So scientists at the University of Cape Coast, for example, specifically, I would want to mention the name Prof. Aaron. So if you come to the molecular biology and biotechnology lab, we have a professor called Professor Aaron. He was able to, champion the breeding of striga-resistant cowpea genotype. And this striga is it's a pathogen. It's a serious pathogen for cowpea. So the parasitic weed, which is the striga gesnesteroids, can cause the 80 to 100% yield loss in cowpea, and that is alarming. So in dairy farming, for example, recombinant bovine somatotropin, which is the growth hormone, is added as a supplement to cows' diets, and this increases the animal's milk production as well. So Prof has done so well by looking at some gene which is able to, withstand the infection of this pathogen to this cowpea. And he has developed so many cowpea, lines. He has been able to come up with so many cowpea lines, new breeds, which is resistant to this striga pathogen. So improvements of soil is also another point. So biotechnology is being used to enhance the ability of natural soil bacteria to give plants nutrients as well as natural fertilizer via nitrogen fixation. And as you can see, or better still, as you have known, nitrogen is very important and is a macronutrient to the plant. So if you get, a bacteria which is able to maximize inputation, or better still, input of nitrogen into the soil for plant absorption, then it's something that has to be cherished and be researched into. And then the next point is the food safety. So when genetic engineering results in reduced pesticide dependence, we have less pesticide residue on foods. So if we are able to rather use the thuringiensis, if we are able to use the agrobacterium, tumefacin, and the rest to engineer our crops without necessarily using the pesticides, which will leave some residues after they are sprayed on our crop, then I think that must be appreciated. The work of biotechnology must be appreciated, because you are not going to use any chemical. It's just the microorganism that is going to work, instead of using these chemicals that have been synthesized from laboratories. And at the end of the day, they will have effects on your system and organs. Now let's look at modern biotechnology in industrial food processes. Industrial food processes. So industrial biotechnology applies the techniques of modern biotechnology or molecular biology to improve the efficiency and reduce environmental impact on industrial food processes. Now, let's look at biocatalysis. Biocatalysis, I'm sure you did small chemistry, so when I say catalyst, you know what I'm talking about. So any molecule that speeds up chemical reaction. So biocatalysis, we are looking at some enzymes, molecules, or organisms that speeds up, biochemical reactions. So the term biocatalysis is mainly used to describe the application of isolated enzymes in production processes. And these isolated enzymes speeds up biochemical reactions for products to be formed, obviously in large quantities in the shortest possible of time. So let's look at advantages of biocatalysis. So biotechnological production yields purer products and consequently requires less extensive purification as compared to complex chemical production. So reduce energy consumption through the ability of most enzymes to work at room temperature. So you don't need so much energy as far as the use of microbes is concerned. And also, less waste is produced out of these processes by the use of these living organisms, because the use of microorganisms often replaces several chemical production steps with one biotechnological production step. Enzymes, which also occur naturally in fruit ripening process, help to soften the fruit cell wall and to reduce the viscosity of the material by increasing the juice yield. That is from 15 to 20%. To decrease filtration times, the number of times that you need to filter it. And it does up to about 50%. Now, it also clarify and stabilize the cloudiness of juice and juice concentrates. And 4, it also decreases risk of jellification. Now, let's look at examples of enzymes used in food processing. So I have some columns here in colors. So the very first column, which is the 1, you have column 1, you have the food processing, you have the enzyme, and you have the effect of that enzyme. And with the 1, food processing, dairy food processing, you have enzymes like the protease, lipase, and lactase. And what this protease does is it's responsible for milk clotting. Lipase is responsible for cheese flavor, and lactase is responsible for lactose removal from milk. Then when you go to bakery industry, you also have amylase, which is for dough conditioning. Xylinase is for dough stability and conditioning and emulsifying. And then phospholipase is for dough stability and conditioning, which is done in situ at site, not away from site. If it's away from site, we say ex situ. And then glucose oxidase, we have the dough strengthening, and then also lipooxygenase, which is bread whitening. We can go on and look at some other examples as well. In beverage industry, we have the pectinase, which is for depectinization and also mashing. Amylase for juice treatment, that is low calorie of beer. And then beta glucanase for mashing. Acetolactase decarboxylase also for maturation of beer. You have the lactase for clarification of juice and also flavor of beer. Now you also have starch. In starch industry, you have amylase, you have the amyloglucosidase, you have the pululanase. You also have the glucose isomerase. Now, with the amylase, you have the starch liquefaction. The amyloglucosidase, you have the acidification. And then also the glucose isomerase, you have it as functioning to convert glucose to fructose. And then we go to the fermented cereal foods and beverages in Ghana. And I'm sure this one is quite familiar with you guys. And so we are just going to look at few of them, like the pito and the rest. I'm sure when we get to this side, the male will appreciate it the more. I'm sure that some of the female will also enjoy the pito and the peteshi and the rest.
Yes,
Charlotte,
sir.
your hand is up.
Doc. Hello, Doc.
Yes, Charlotte. Please, I can hear you.
Please, the last part. I heard you were saying about pito, as to whether some people, I mean, is a delicacy of some people. Am I right? I think I didn't get the last part clear.
Okay, what I said is that we are going to look at the activity of microbe in production of pito, the ahe, and the rest.
Oh, okay.
The local food drinks, okay, or beverages.
Okay. Oh, okay.
Yes, we are looking at the, activity of microbes, the
Oh, okay.
biotechnological procedures that are involved in preparation of these local drinks.
Okay.
All right.
Thank you.
Okay. So can we continue?
Yes, Doc.
Okay, so fermentation, all this begins with fermentation. These are fermentation processes. And we know that during respiration, if there is no enough oxygen after the oxidation of glucose, if there is no enough oxygen, then the fermentation process results either in production of lactic acid or production of alcohol. If there is enough oxygen, then Krebs cycle will continue, and then also the energy molecules will be fed into electron transport chain. But In this, our learning or studies, we are going to consider the fermentation process. That is when there is no enough oxygen. And so bacteria, there are some bacteria that really work under low oxygen conditions. And so we are referring to them as anaerobic organisms. The aerobic organisms work in oxygen abundant or oxygen efficient environments in producing energies for tissues and cells. So yeast is also, an organism that works in very low oxygen concentration environments. That's why we use them in bread making and all that. You can even use it in preparation of akpeteshi. Right. So let's look at fermentation. Fermentation is the use of desirable microorganism in the production of value-added products of importance. So fermentation occurs in nature in any sugar-containing mash from fruits, berries, honey, or sap tapped from palms. If left exposed in a warm atmosphere, airborne yeast acts on the sugar to convert it into alcohol and carbon dioxide. The making of wine and beers uses this biotechnology under controlled conditions. So, all these procedures are biotechnological procedures. So, let's look at health benefits from fermentation processes. Now, fermentation processes increase nutritional level of food. For example, vitamin B12, folacin, and riboflavin are all increased after fermentation of maize flour. So, the fermented grains and vegetables are full of the beneficial lactic acid by bacteria. Fermentation of cereal leads to improvement in digestibility and significantly lowers the content of anti-nutrients of cereal products. And I have to be quick to add that there are some, components of cereals that are not digestible by human guts or the enzymes in humans. For example, cellulose. Human beings, we cannot digest cellulose because we don't have, enzyme for digesting cellulose. So it's the bacteria that does this digestion for us to release some very, very important vitamins and fatty acids in these cereals. Now, they do this under anaerobic conditions. So, yes, we will still talk about fermentation. And it also improves preservation, that is fermentation. It improves preservation, converting perishable food into the one that has a longer shelf life. Fermentation changes the nutritional value of food products. Example, converting milk to cheese. It can create or improve sensory characteristics of food, such as the taste, flavor, aroma, and texture. And you can agree to me that in our household, whenever they send you to go and mill corn, they don't use it the very day that you bring the corn, or better so, just after milling the corn. They would want you to put your hand in there and mill it, maybe mix it with water, the flour with water, until you get a uniform consistency, and you keep it there for some time for it to bloat up or some bacteria to act on this milled corn and produce some, interesting flavor, and then you use it to prepare your banku for it to taste well. If you prepare the banku just after, milling the corn, you wouldn't get that taste as desired by everyone. But then that characteristic taste that is achieved is through fermentation process, as when the corn or the dough is left for a day or two. It can also create or improve sensory characteristics of these taste and things that I have just mentioned. Let's look at some products from fermentation process. And we can have palm wine. So the white liquid or initially collected tends to be very sweet and non-alcoholic before it is fermented. And in our local dialect, we call it insefu or insafufu. I don't know how in your local dialect they call it. Yes, and then insefu, and we
Yes.
enjoy it so much. But when you live in insefu or insafufu or insefu, there for some period of time, you have the bacteria or some yeast components digesting the carbohydrate content of it under anaerobic condition to produce the alcohol that results from it. So within some 2 hours, fermentation yields an aromatic wine up to 4% alcohol content, which is mildly intoxicating and it's quite sweet as well. Right. So we can also talk about pito. Pito is a sorghum and it is soaked in water for 24 hours and made to sprout for 3 to 5 days. It is then dried at temperatures between 25 to 37 degrees Celsius for 24 to 48 hours. And they mill it. And the dough is mixed with water using, sometimes they use okra stems and they allow it to settle. And the upper part, which is the supernatant or the extract one, is collected in a bowl. The supernatant is boiled for about 45 minutes and they cool it. And pito is fermented by back sloping. Thus, we inoculate the extract one with a part of previous extract two containing yeast and left for 24 hours. So the one that you mill, they rather use the previous one that you have, the previous pito that you have prepared that contains the yeast, because yeast, they are organisms that are still multiplying and they have short generation time, as in they multiply very fast, and so they can reproduce and undergo anaerobic respiration to produce some characteristic flavor, which is production of alcohol, which has your pito taste, and that is called the back sloping, or you can also say inoculation, introducing the second extract into the first one that you have just prepared and leaving it for 24 hours. We also have a popular one called ahe. I'm sure I pronounced it right. Ahe. Please, it's not ahai, it's ahe. Yeah. So it is sweet. Yes. I think I just took some this afternoon when I was coming from town. I bought some and I took it. It was nice. So ahe is sweet, especially when they add milk, a bit of milk or a pint of milk to it. And it's it has some malty tasting beverage. And it's brewed from maize in southern Ghana and is usually served as a welcome drink at outdoor ceremonies, waykeepings, and some funeral events. The traditional method for preparing ahe is much the same as for pito. And the acid alcohol beer brewed from the sorghum or millet in West Africa, except that of ahe,
Hello, sir.
is not
Sir.
boiled again after fermentation.
Hello,
Yes.
dog.
Yes.
Please,
Bernadette,
what is the local name for the ahe?
the local name for what? Is what?
Okay, okay.
I think the
Okay,
one is called
thank you. Thank you.
Ah, So, oh, so you mean it's not a popular name, eh? Is that what you are implying?
Yeah, I think it's.
Yes, we call it Emiejo.
Ah,
Or
you people. Oh, okay. Sorry about that.
Ah,
I didn't know it's not a popular name.
thank you.
Oh, okay. So you
You
call
also call it
the guns call it Emiejo. Is that right? Is that right? So the Ewes call it what? How do you call it in Ewe?
Aleha.
Aleha. Oh, okay. What about the northern sector? Do you have a popular name for it?
I think one popular name
sector,
of it,
we don't have it.
Oh, okay.
I think it's popularly known as Asana.
Asana. No. Asana is different. No.
Okay.
Asana is different or is the same?
Yeah,
yes, some people call it Asana.
Only the asana they put salt inside. When you come to Ghana area, they put
I know
salt
Asana is different. I've tasted Asana before. It's not the same Asana.
Yes, they put salt, but for Aleha, they don't put salt.
Yes. All right. So I think we can continue. So this is this is a popular dessert which is found in the southern sector. It's a popular dessert which is found in the southern sector. All right.
They sell it in gourds.
Yes, they sell it in that this thing.
Okay.
Now they even sell it in a rubber container, in a plain rubber container with some ice cubes in it.
That's Asana.
That's it. That's Asana. I just googled it and that's the same name they are giving me. It's either called Asana or only one the Leha.
Okay.
So it's the same thing.
Okay. All right. Right. So I'm sure some of you are familiar with this, and please be sure to have a taste of one. Since appetition is not helpful, you can resort to that one. I'm just advising my brotherhood.
Then Sanfo is okay.
Ajimai, hear it.
All right. So I think we can go on and look at the kenke. Kenke, how to prepare kenke to. It's a biotechnological process. We still introduce microbes to get that characteristic taste, both with the Ga kenke and then the Fanti kenke or whatever kenke that you prepare from your ethnic dialect or your ethnic line. Let me just put it right. So kenke is a popular food prepared from fermented corn dough, and it is semi-cooked in a hot water into a smooth whitish consistent paste, after which it is rubbed with corn husk or plantain leaves, giving it different texture, flavor, and also a color. They are boiled for a long period of time into a consistent solid balls. But before these are boiled into these consistent solid balls, they would have to be fermented so that the color and also the taste would be different from one, who embark on this from another individual. So the way the Fantis do their own is quite different from that of what the Gans do. Not entirely different, but then the Fantis I learned they use the plantain leaves to do it, and that of the Gans they use the husk, the maize husk to do it. I'm sure this time we are having different ones also from our sub-Saharan African countries. Different one. Okay, we have different one coming up from these sub-Saharan Africans as well. They don't use the husks, others also don't use the plantain, they use different mode. But in essence, what it means is that if you use the plantain, it will give you a different taste. If you use the maize husk, it gives a different taste. But essentially, the technology behind the preparation is a biotechnological process, and that technology is the fermentation process. Okay, it involves the use of microbes or introduction of microbes into the dough, and later prepared, or better still, temperature is added to kill these microbes that you introduce into the dough. And you can have your your kinky boiled into the solid balls that we normally see. Let's look at some other foods as well. We also have banku, or the akpele. I think the akpele is associated with the West, and the banku is associated with the Northerners, the Fantis, the Ashantis, the Nzumas, and the rest. I don't know whether the northern line, do you have different banku apart from?
We have the
Hello,
TZ.
sir.
What about the
TZ.
etil?
Yeah, the etil 2 years. I think the etil has metamorphosed into banku, because me, I grew up knowing etil
Hello, sir.
all along. Yes, sir. Prosper, your hand is up.
The akple you are talking of, is it the powdered one? Is it the workplace or the banku?
Hey, brother, you are now coming to
Hey,
confuse me already.
Because we have
The
the
akple,
workplace, that one.
do you have types of akple?
That, yeah, the one we use the corn flour, that is the workplace. But
Okay.
the banku in general, we do, yeah. Okay, that's what I'll say. Mostly it is the workplace that they've been calling akple.
Okay, so which one is the akple?
The one they use the corn flour.
The corn flour.
Yeah.
Okay. So whether the corn flour or the workplace that you are talking about, you still, introduce microbes into it. Is that not true? You introduce microbes into it with your hand.
Well,
I mean, you don't
with
mean to introduce a
with
microbe by using
with
yeast or
with
something like that.
with the
You eat them.
workplace, it is the maize dried one that is grinded into the flour. So that
So,
one they don't ferment it.
mhm.
But with
But
the
at the end
banku,
of the day, you don't use it
uh-huh.
right after grinding. Is that not true? Or after milling? Is that not true?
Oh, you can use it the same day.
Oh, really? And it to give you that characteristic taste that you need.
Yes, please.
Oh, okay. That's fine.
That's the that's the
That's the
work
actual.
that's the workplace.
Aha. So we are talking about the aquilent rather, right? So it's not the aquilent. It's not the workplace. It's the aquilent that we are talking about. Yes, Emmanuel, your hand is up.
Sir, please, so the Everest, but if it is banku or akple, they or the workplace, they refer to as akple.
Okay.
So when you put the akple there, and then one can call banku akple as well.
Okay.
Before we have the workplace.
A workplace.
So when you say akple, but for the after the airways, either akple or banku is the same thing. They call it the same thing.
Thank
Okay.
you, bro.
Okay. That's fine. Because I know some joints at Pedu Junction, Cape Coast Pedu Junction, that have been buying aquile there. It's just like the bangkong that have been taken. So as my brother Prosper Mate was saying, I was kind of confused because I was getting almost the same taste as bangkong. Yes, sir.
The difference is that with the what Prosper was mentioning as a akplen, that is the original akplen, and then they have modified it. Some of them add what they call the cassava dough.
Cassava dough.
So that one
Okay.
is that's not the original akplen. The original akplen is the one with the corn flour alone. But
Okay.
the moment they add what they call the cassava dough to it, it has changed. But they call all of them the same thing. But the bankua for banku is different from all this.
All right, all right, all right. Fantastic one.
I just ate akplen, so I know precisely what I'm talking about. And that one, it doesn't even undergo fermentation. Oh. You can grind today and use today, or
Is,
for preservation methods, you can just put it down. It doesn't undergo
is that
fermentation.
the akpele or the wokpele?
The akplen, the akplen. You see
Akpele.
the
Hey, don't confuse us.
akplen, the akplen is
Everyone
the
is
same.
bringing the version.
No, no, not the version. You see, the moment they add the now they have modified by adding the what they call the cassava dough. And the moment that one is added, they still call it akplen, but that is not the actual akplen or the wakplen we are talking about. That one is different, and that one is the one that look like the benku.
Okay, so what's the name of that one? So that one is called what?
We also call it akplen, but the name just came out of because they use the corn flour to do it. They don't have any
Okay.
precise name.
What
That's
them
the
is?
second.
What them is?
Hello,
Before you
sir.
come,
The,
what them
the,
is?
the, the thing is, the
I think
flour
it's the
is what
same
they
for the
call.
Etel and the Banku. The Banku
Okay. So all right. So when it comes to
and
the southern sector too, okay, they some call it etil, some call it banku. Etil, we don't add any cassava. Cassava dough. But with banku, you just add some pint of cassava dough to the corn
Hello,
dough.
dog.
Then become the etil.
Hello, dog.
Yes, Bernadette.
Local people are saying Akpele don't go under fermentation.
Uh-huh. So
Thank
we
you.
should take the akpane off and use our banku. All right.
Yes, please. I'm sure that's it.
Right. So these are foods prepared in Ghana from fermentation, or better still from fermented corn and cassava dough mixed proportionally, and is cooked in a hot water in a smooth whitish consistent paste, and they are folded into balls after they are cooked, and they are served with soup, sometimes with stew, or with pepper sauce, and also with fish, maybe shrimps or tilapia or something like that. Okay, right. So on the note of akpale and the wokpale, I think we can move on and look at the fish products. So we also have the stinking fish, which is the momone. We also have the kobe, and the stinking fish, that's the raw fish, is either left overnight before salting or is dried, salted immediately after washing. Salting and fermentation lasts for 1 to 6 days, after which the fish is dried on ground, either on grass, either on earth, or on stones, or it is raised on a platform for 1 to 3 days. And sometimes we have the kobe too, which is the raw tilapia fish, and it is dressed and thoroughly washed. Now you have the dry salt or dry salted ones is rubbed and it is grilled sometimes. Now these gels that are salted are exposed to some form of temperature, the sunlight, for drying, and they are arranged in alternative layers with salt, and they are allowed to ferment the more for 2 to 3 days before being dried for 2 to 4 days. We also have the fermentation processes in beer making, and it is probably one of the oldest beverage that have been intrinsically linked to biotechnology. Now, Opoku Juliet, your hand is up.
Hello, Doc.
Yeah,
Please, I
Julie.
want to ask this. Please, is there any negative effect on feeding on this momone, Kobe? We just learned from you.
Right. So um you know that your body needs some macronutrients, and the macronutrients include sodium and chloride ions, and that is what you use to preserve your Kobe and the momoni. The macronutrient, which is the sodium and chloride ions, are used for transmission of nerve impulses, but they are also used in regulating your body metabolic activities like your water solute balance in the body. Now, if you have excess intake of these ions, okay, you have deposits of these ions, and they will go a long way to affect your systems. That notwithstanding the negative effect, this momoni and then the Kobe we are talking about, some people use for it not to go bad. Some people, the market women, for more economic gains and greediness, let me use that in quotation marks, in greediness, they use formaldehyde in preserving them. Now, formaldehyde or the formalin is a carcinogenic agent. And what I mean to say is that they cause cancer. They cause cancer. So if you go to the market and you are buying momoni or Kobe, be sure you go to the market when who has a lot of flies hovering around his or her Kobe before you start patronizing. If you have a market woman who has no house fly on his or her Kobe or momoni, please run away for your life, because these house flies are sensitive to these formalin or formaldehydes. Right. I have Income and Charlotte. Your hands are raised.
Yeah, Doc, please, on the protein, the fish,
Great.
I stand to be corrected. The fermentation, the sign to, so one of the signs, the sour taste.
Okay.
After that sour taste that is produced or
Yeah,
detected or taste,
the characteristic taste
yes,
of
of
momoni
the
and fermented
but in
products.
this case, when you take the momoni, it doesn't. I don't know whether how the fermentation is done there without that thing, or is it because there are protein substances? That's why you don't detect the sourness after the process.
It
Process.
depends
I want.
how long. It depends how long you expose or you leave it for this fermentation process to proceed before salting it. Because just after salting it, okay, just after salting it, you stop the activity of these microbes. Just after salting it, you stop the activity of these microbes that we are talking about, because these microbes cannot, inhabit salty environments. So just after salting it, you stop the action of these microbes. So if you leave it for a long period of time, these characteristic states that you are talking about will be detected. But just as you bring it from, the water body, you dry it small before you salt it, you may not get the characteristic state that we are talking about, because fermentation process did not really pass. Please, did you get what I said? So if you really need the
Yes.
characteristic taste that we are talking about, you have to leave it for quite a number of period of hours before you salt it.
Okay, though. So in this case, how will we? Put when we say to putrefaction. I don't know. Is it connected to this fermentation?
What?
To putrefy. I don't know. No, but I've checked in some I read in this somewhere, and they said when the protein is going bad, it putrefies. So they were saying that make it, like, for instance, the production of more money. That is, they allow the descent to. So that is why I'm a bit confused. I don't know whether that word term to putrefy or putrefaction is connected to fermentation, or maybe fermentation is a general term. I'm a bit confused. That's why I'm asking this question.
Okay. So when you say you want to putrefy it, okay? Can you hear me?
Yes, Doc. Yes, I'm listening.
I think there are some individuals who have your microphones on.
Madam Beatrice.
Hello?
Hi.
Hi. Hi, we are on.
Nobody
Why
should call
is
me
that
to
my
tell me
dog
to
is
call him.
up?
Oh.
Why? Yeah, you go and talk now. Dog is up.
It says Doc is off. Oh.
Mr.
Nobody should
Ademai,
tell me to call
text
him.
him
Please,
message.
I'm not going to call him. No, he
Text
will
him
come back.
message. Text him.
Yeah,
Message
Mr.
I
Jemai,
saw.
He will
test
come back.
him,
He will
test
come. Don't
him.
worry. He will come.
he will
Now
come. He
even
will
tired. He
come.
will
Mr.
come.
Ademai, do your job.
Nana
Aunt Mercy, if
Ajema.
you are around, do your job.
Ajema Papa be. Text him, okay?
I don't have data to call him. I don't have data to. So let's wait. He will come.
Text
You
him.
have data to join the lectures.
Text him. Oh. We beg you.
Yes, I have two different data for different operations. So wait for him. He will come.
Text him for us, so please. Aunt Mercy, if you are also there, text him for us.
If for Robert, your face with that. You, eh?
Very good.
Hey, please, is dog network off?
Prof, they
Or,
go off. Oh.
or is
Doc,
break?
they go off. Oh.
Ah, okay. Hey.
Prosper, continue from where Doc left.
It's all because of purification.
Oh man, what is what is wrong with that? Ya bre. Wait there, ya bre.
You know, somebody way equals to
Prosper.
Hello.
Prosper.
Hello.
What they go? I will bring.
Please, the recording is always on. Oh. Though we are always now playing on the platform.
you know, this is the way.
I want break or
Hello.
dog
Hello, please.
is listen.
Hello. Yeah, please. Yes. So, in as much as he is off, let us try and exercise a little bit of restraint. You see, at the end of the day, you play back these recordings and you all listen. So let's minimize the laughter and those kind of things. I've tested that he's off. He has not responded. I'm waiting for his response. When he comes, we all see. Okay. I'm not going to call because he said we shouldn't call. So I'll send him a text message. Let's hope that he'll reply and know the way forward. But by the way, my team, United is flying high. That is why you've not heard my voice. I am not coming to talk on the platform. Whatever we discuss, I'll contribute. Thank you.
Thank you. You're welcome.
Okay, love is back. So let's follow.
Yeah, hello.
Hello, Doc.
Yes, sir.
Hello, Doc. Good evening.
Great. So there were some calls coming in from my side, so I lost my network.
Okay.
Sorry about that.
Oh, no.
Okay.
Yeah. Let's go back to what we're discussing. So beer making. Was there any concern? Was there any concern?
No, sir.
So it was my now this thing, I wanted to get whether there are other forms of fermentation. That's what I wanted to know, whether in protein we have a specific name or the fermentation goes for both protein and then carbohydrate based substances.
Okay, so you are talking about maybe the composition of, or better still, it comes back to the use of these microbes. Okay, yeah, you know that in all living organisms there are carbohydrates present, there are proteins present, there are vitamins present. These organic molecules are present in almost all organic compounds, right? So when you are interested in maybe decomposing these protein components of organisms, like in the case of this Kobe and then Momone and then the rest, fine, you are decomposing, or better still, you are looking at respiratory activities of these microbes occurring in them. Like I said, you introduce these microbes into these bacteria, into these organisms, like the Kobe, tilapia, and the rest. And then you allow it some hours for it to, ferment before you salt them to cease the activity of these microbes. That notwithstanding, when it happens like that, there is this decomposition of other organic components, like the protein components, like the vitamin components, like the fat components, and all that. But then the decomposition of the protein components that give this characteristic smell. And then a bone, that's when normally you just do I say forbid the smell. You wouldn't want to get in touch with this even in your dress and all that. This foul smell that comes off of this decomposition process is what normally we refer to it as the petrification.
Okay.
Uh-huh. That taste, that characterizing foul taste or the smell that comes out of it.
Okay. So that means that do we classify that as fermentation as well?
Yes, it's a fermentation process.
Okay.
Yeah, it's a fermentation because living organisms are involved. Living organisms are involved. Is that okay? And
Yes,
it's
Doc.
it's an anaerobic activity. It's an anaerobic activity. All right.
Thank you.
Yes, Julie, your hand is up. I can see Julie's hand.
Yes, sir. So please, if I should understand, it means that when this tilapia is decomposed, I mean this Kobe, it means when we feed on, there is a kind of nutrient we obtain.
Oh, we are saying that after decomposition or better still after fermentation of these processes or these compounds.
Hello.
Can you hear me? Good. So after
It's me, my network.
okay, so after fermentation of these compounds or better still after decomposition of these compounds, it doesn't mean that the organic components of these particular organisms are entirely destroyed. Okay, but there are is exudation of some very important vitamins. For example, in the case of cellulose, if cellulose is decomposed or better say is fermented, it doesn't mean that because the cellulose is destroyed, the chemical structure or better so the chemical composition of the cellulose is reorganized. It doesn't mean you don't have any compound which is beneficial to the human or better still to the organism. Like I said, the reformation of the chemical structure of cellulose will yield certain very important elements or compounds like vitamins or fatty acids and other compounds that are important to the health of the organism. So reorganization of these molecules, okay, doesn't mean that the organic components of the this Kobe and things are entirely destroyed. No, you will still get your nutrients, but maybe in different form. You may not get the protein components of it, but then since it is digested, these molecules are broken down into simpler forms. You will still get the nutrients which you're supposed to get. Is that okay? Despite the gases that are released, despite these gases that are released after their decomposition or their degrading processes, which is foul to you, which is not really good to you, you get the nutrients that you are supposed to get.
All right. I want to break some misconceptions people have about feeding on these things. Thank you, sir.
Yes. All right. Either than that, then Ghana Standard Authority would have bound, or better still, would have just called off the eating of these food, components from the markets.
All right, sir.
So
Thank
let's then proceed and look at some other form of fermented products as well. So wine, I think we have spoken about the beer. So most of the
No,
processes
please.
involved in beer making are enzymatic. Different. Am I gone again? Please, am
You're
I
on.
gone again? Hello.
No, no,
No,
it's
sir,
still
you're
there.
on.
Hello, please, am I gone again? Am I still there? Am I still with you?
Yes,
Yes,
please.
please.
We
No,
can
yes,
hear
You
you.
are
please.
Yes, yes, yes.
Okay. All right. Right. So the most of the processes involved in beer making are enzymatic, which is different from types of beer, and they are made by using different ingredients and also different production techniques. The main strains of yeast used for fermentation are the Saccharomyces cerevisiae, which is the yeast. I mean, they are the different variants of yeast organism. The Saccharomyces ovarum and then the Saccharomyces cerevisiae. They are the different variants of yeast. The distinct steps in beer making are malting, mashing, sparging, boiling, fermentation, and then also packaging and aging. We go to the wine making. So the wine is produced from fermentation of fruit juice, usually from grapes. And there are 5 basic steps to making the wine. And it goes on to say harvesting or picking, crushing and pressing, fermentation, clarification, which is the sedimentation, and aging and bottling. The extracted juice is then treated with sulfur dioxide to kill bacteria and naturally occurring yeasts. But it also helps prevent oxidation, of these free radicals. Then sugar content and pH are adjusted before adding a starter culture of yeast, usually a variety of Saccharomyces cerevisiae. In other words, yeast. Then we come to yogurt making. So yogurt is a food produced by bacterial fermentation of cow's milk, whose constituent protein is mainly casein. So casein during digestion, casein is acted on by the proteases because casein is a protein and it is converted from carcinogen to casein. Now, in making yogurt, the milk is harmonized or homogenized and pasteurized. Pasteurized is application of temperature to, end activity of some microbes, bacteria, and the rest. Fermentation then follows by adding a starter culture of bacteria, usually a mixture of Streptococcus, Haemophilus, and Lactobacillus agaricus. Okay. These bacteria act on the milk sugar, which is the lactose, to lactic acid, which coagulates the milk protein, casein, to produce the thick, creamy consistency of yogurt with slightly sour taste. So the sour taste that you mostly feel when you take in yogurt is the taste of lactic acid. All right, so the fermentation works best at temperature of 46 degrees Celsius, and when complete, the yogurt is cooled to 50 degrees Celsius to stop the bacterial processes. Some kinds of yogurt contain probiotics, and probiotics are live bacteria and yeast that live inside human digestive system and are good for human health. Example is the Escherichia coli, normally referred to as the E. coli. They help in digestion as well. Right, so benefits of probiotics. Now, it decreases potentially pathogenic gastrointestinal microbacteria or microorganisms. Now, reduction, they also reduce gastrointestinal discomfort like belching and the rest. They also strengthen the immune system by providing us with some very important vitamins, like I mentioned, digestion of the cellulose. Now, they also improve movement regularity. In other sense, they prevent constipation. And they also maintain individual intestinal microbiota in patients receiving antibiotic treatment. Now, we continue by looking at some local foods produced out of fermentation processes. And the 10th one that we are looking at is bread making. I'm sure I haven't been lost.
No, sir.
No, please.
sir, you
Yeah,
are on.
Cosre, please, am I on?
yeah, on.
Am I
Yes,
come
you are
again?
online.
Please, you are online.
Okay, okay, all right.
Yeah,
I think some calls have been coming in. Some calls, maybe going forward, I would have to get a separate phone for this presentation procedure if I'm not on campus, so that it will save us from getting me offline whenever I'm presenting. Sorry about the inconvenience, please. Intermittent distractions. Right. So please let's continue. In bread making, cereal grain is crushed to form flour, thus exposing the starch or the stored starch. Water is added to activate amylase in the flour. And you know, amylase is an enzyme that acts on the carbohydrates, specifically starch. So yeast Saccharomyces cerevisiae is added, which uses the glucose as a respiratory substrate, producing carbon dioxide, which forms small bubbles trapped in the dough. Upon baking in an oven, these expand because carbon dioxide has been produced. These gases have been produced out of the anaerobic activity. And so it bloats the bread, giving rise the bread in a light texture. Some anaerobic respiration, nevertheless, takes place and the alcohol produced is evaporated during the baking process. Now, let's look at modern biotechnology in medicine and healthcare. Now that we've ended the local foods that are produced, I'm sure if you are asked to mention and explain some few fermentation processes involving some local food products, you wouldn't have a difficulty at all in mentioning some few for us. And you will not come back again and tell me that I did not give you sample questions to try your hands on. That is on the lighter side. Please, as we move on, kindly put down some few questions for yourself. Aside you waiting for me to give you some sample questions, which I will not fail on doing that. But just in case I fail on doing that, then you have prepared yourself in secrecy to give me an A score in biotechnology. Is that okay?
in fact, sir, you will not fail. You will do it.
Sir,
It will not
for
be the
every
same
action,
person.
there's equal and
You
opposite
can't fail
reaction.
us.
Okay. So in as much as we are
All
putting in
right.
our effort, and then effort from your end too will help us to get the A.
But it's easy.
Well,
I mean, it's easy getting.
for the past three terms, Dr. Abuaje's course, you've all been scoring A's and B's in it. So you are rest assured. So to every reaction, there's equal and opposite reaction. So that is how it's supposed to be. Thank you.
Come again. You said something which interested me,
What
or
I'm saying
which
is for
interests
every
me.
action, there's equal and opposite reaction.
Because I'm getting a brace in your what you're saying.
That please is saying for the past
Is it from my
semesters,
end?
Dr. Boaji's courses, we've been getting A we've been getting A's and B's, so you are assured of the A's and the B's. But by then, yeah, you also help us with some questions, tutorial ones. Then, we go blow the A's for you, dog. Don't worry about the A's and the B's.
Oh, okay. Okay, that's what you mean, right? Oh, okay. Oh, no, no. In as much as you are looking at getting the A, you have to work for it. That's what I have to tell you. You really have to work for it.
Yes, sir.
Yeah.
Sir, this one
Please
is
work
like we are
hard.
going to write more before exams.
Before you do
Sir,
what? Mm-hmm.
it's like we are going to write mock before exams. So the questions that maybe you give us is our mock to guide us, so that before we can write the exams well.
Sure. Oh, I trust you guys. I know you will do it. And it's not something that is going to be quite difficult. If you don't learn, then it will be. I told you that nothing is coming out of these slides. Everything that I'm giving you, everything that I'm teaching you, that is what I'm going to bring. I'm not going to bring anything outside what I'm teaching you. That will not happen. That will not happen. So please, if you study the slides, you pass. Let's focus on regaining the knowledge. Knowledge gaining is very, very important. Okay.
Sure,
The gain
dog.
of
We
knowledge
are ready
for you
to
to also
gain the knowledge.
transfer is very important. That is why I most of the times pause for you to ask questions to clarify what you don't understand and the rest. So please, knowledge is also very important. Don't only focus on the grade and the grade and the grade. Grade is also good and important, but please, at the end of the lesson or at the end of the course, you should be able to at least also be able to, transfer your knowledge to others as well. So that's very important. So let's go to modern biotechnology in medicine and healthcare. So medical biotechnology include vaccines, monoclonal antibodies, gene cloning for medical purposes, cell and tissue engineering. Number of antibiotics are being produced by fermentation or in cell-based systems. So medical uses of modern biotechnology fall in two domains. One, testing or diagnostics, and two, prevention, control, and treatment purposes. Now, let's look at the testing or the diagnostic domains. So, one, accurate diagnosis of a particular condition of a disease or disability is key. The prediction of a disease or disability in a person that will lead to the development of certain symptom is also key. Now, you test for susceptibility to a disease or disability in a person, and you test for career status of inherited condition of offspring, testing for a career status of the inherited condition of offspring of a genetic trait. So you use it both in testing and the next, which is the prevention and treatment. Now let's go to the prevention and treatment with the biotechnology. Now that you know that the condition is being inherited by an offspring, or which is transmitted from the parent to the offspring because of the offspring carrying a gene sequence from the parents, what can you do to help these offspring? That is prevention of the manifestation or expression of the genetic traits by the offspring. One is you can do gene therapy for the individual. Now, this gene therapy involves introducing a new gene into the person to treat or correct the incorrect expression of the gene, and also stimulate an inactive gene or restrict an overactive one, All these have been explained when we're doing genetics, as in the very basis of, trait transfer. When we're talking about transcription, I spoke about that. It has the potential to correct conditions that affect the brain, the central nervous system, which is the Parkinson's disease and Alzheimer's disease, and incorrect gene expression that leads to intellectual disability. So gene therapy has the potential to treat cystic fibrosis, heart disease, HIV and AIDS, diabetes mellitus, and also cancer. So this is what gene therapy can do. That is correction of a gene sequence, or to correct a gene sequence so that it doesn't produce a protein which will manifest in the manifestation, or better said, in exposing the individual to certain ailments that is associated with genetics. Yes, Frank, please, your hand is up.
Yes, Doc. You said it has the potential of treating HIV and AIDS. Yet we still
Yes.
have.
Yes, we have it's associated with your gene sequence. Now, when you are infected with a virus, okay, when you are infected with the virus, what happens is that your gene sequence is being changed by the viral gene so that proteins that you are going to produce will be useless in the body. When these genes are connected, or the gene sequences are corrected, you start producing your normal, proteins that are responsible for coding for specific characteristics that initially you were producing. So we can use the CRISPR-Cas. Okay, that is a new technology for gene screening or gene correction or the gene therapy. The CRISPR-Cas. You can read around it. And how the technology works.
Doc, in that case,
so they can use that technology to correct.
Or it is an expensive venture.
It's what?
Do you have people who are being treated from this HIV/AIDS, or just that it's expensive, or is something that is in the pipeline?
Yes, what happens is that yes, we have some people who are treated. And the symptom never manifested. You have some people who are infected and they have been treated and they are well. They are not showing the disease at all. They have been healed, but it's expensive, just as you put it. Gene therapy is not for the weak. Gene therapy is not for the weak. It's very expensive process, but it's possible in correcting this cancer. What has been stated, the cystic fibrosis, and then all that. But it's expensive. If you have the money, you'll be healed. It has to do with correcting the gene sequence. Those portions that are useless in producing those nonsense proteins will be cleaved up.
Hello, sir.
Yes, sir. Yes, sir.
Yes.
Hello, sir.
Yes, sir.
Yeah. So my question is, this morning from the molecular genetic class, we made aware that cancer, when the gene is affected with cancer, you stays reduced.
Come again.
And here's the case you're also talking about. I said this morning
Come
during
again.
the molecular genetic class. Hello.
Hello.
What I'm saying is, I said this morning during genetic molecular cells. We're made aware that
Okay, molecular genetics.
cancer,
Yes.
when the cancer is when the gene is affected with cancerous cells or cancer, it is not
When the what?
treatable. You can't treat it.
When the
What I'm saying
gene is
is we're
affected
made
with
aware that
cancerous cell.
yes, you
You
can't
mean
treat it.
when the gene is affected with cancerous cells?
He said he said that genetic mutation, when it mutates and then the gene gets cancer, you can't treat it.
The gene
And
cannot
I'm
get
asking,
cancer. The gene
no, so
cannot
he
get
said that
cancer. It's the cell.
what I'm saying, I said
Okay.
during the class, he said that when you are affected
Uh-huh.
with cancer, when you get cancer, you can't
Uh-huh.
treat it. Cancer
Yes.
is not treatable. But here's the case, you're talking about gene therapy that we can also use it to correct HIV and cancer. That is where my confusion is coming from.
So where is the chemotherapy coming from in here? If the cancer cannot be treated, do you know that they can put you on chemotherapy? Systems, and then you'll be cured.
I know.
Yes, sir.
Do you know that they can also do radiotherapy for you?
Yes, I know of that one.
Yes, it's also
Yes,
there.
though.
Yes.
If it's detected in early stages, if the listen outgrowth is detected in early stages, you can be treated.
Okay.
You can be treated.
Hello, sir.
Okay.
Hello, sir.
Yes.
Please, this morning, I think the emphasis was
Uh-huh.
the cancer. It was, I think, leukemia in the bracket. And the doc emphasized that no amount of therapy can bring you back to life. You die, no matter how therapy they put you on, because leukemia. I think the emphasis was on leukemia. So if Mr. Ademai can remember that portion, I don't think it was
Yes.
Okay.
always
So, and
on
what happens
food.
is that it depends. It depends on the extent. It depends on the extent of damage. Okay. And if it is localized, the cancerous cell is localized, it can be treated. But if it starts spreading, that is where you get the problem of treating it. If you are not careful, you even destroy the living tissues, especially when you subject the individual or the patient to radiotherapy or the chemotherapy. You can destroy certain live and functional tissues. And that is where we have the problem. But if it is localized and it's at the early stages, it can be treated. Please, do you get it? Hello?
Yes, sir.
Akomdai, do you have your hand, please?
This gene therapy, is it possible in Ghana?
Come again.
I'm saying the gene therapy, is it possible in Ghana?
Oh, most of the times. Oh no, they have to fly you outside. For gene therapy, they have to even gene sequencing. Gene sequencing is this late that we have some of the equipment available. So with the gene therapy, they have to fly you outside.
Okay.
Akumdai, did you get that?
Okay, thank you, sir.
Yes. Yes. All right. So we are talking about the change in sequence of the DNA, the genetic structure. And we don't have to confuse ourselves with the gene becoming cancerous. Cancer has to do with the cell, okay? Uncontrolled multiplication of the cell. Now, when we talk about gene therapy, we are interested in the sequence of the arrangement of the genetic material. That is what where our interest is. It's beyond the cellular level. It goes beyond the cellular level. Now, before a cell multiplies, there should be a duplication of the genetic material. There should be a duplication of the genetic material. So, if you have duplication of the genetic material and these gene sequences have been corrected, the faulty or the mutated ones have been corrupted by this technology that I refer to, the CRISPR-Cas, then you are going to have proteins, or better still, you're going to have, duplicated gene, or better still, duplicated DNA or genetic material, which is going to code for, characteristics that is desired, or better still, characteristics that are functional in the body, and no more the production of proteins that are not useful or useless proteins in the body. That is why we say that with the therapy, you can correct the sequence of the gene that has been mutated, and with the mutation portions that are multiplying in forming the cancerous cells that we experience. So with cancerous cells, we can say that the gene has become cancerous. I think one individual made mention of that. We don't have a gene which has become cancerous. We have a cell which has become cancerous. So kindly take note of that. Right, so with this therapy, you can correct the sequence of the gene. We also have approaches that are involved in the gene therapy. So a normal gene could be inserted into a non-specific location within the genome to replace the non-functional gene, just as I just said. An abnormal gene could be repaired through selective reverse mutation. Now, regulation, that is the degree to which gene is turned off on or on of a particular gene, could be altered so that the gene cannot express to produce any protein. You mean the abnormal one can be turned off so that it doesn't express to produce any abnormal proteins. An abnormal gene could be swapped for a normal through gene homologous recombination. Now, I'm sure that with your molecular genetic courses or the time with a molecular genetic teacher, you were taken through the cell cycle. And during the cell cycle, you came across a process called homologous recombination and then the crossing over. I'm sure you are familiar with these terms. So let's look at some of the problems of gene therapy. The risk of stimulating the immune system in a way that reduces gene therapy effectiveness is always a possibility. Now, another thing is that viruses being the carrier of choice in most gene therapy may recover their ability to cause diseases once inside the patient. Now, also, multi-genetic disorder could be especially difficult to treat effectively. You know, some diseases are, influenced by multi-genes. They are controlled, as in they are controlled by different genes or different alleles. Let me just put it right. They are controlled by different alleles. So it will be quite difficult to treat it. The virus may target the wrong cell if the DNA is integrated in the wrong place. That is also another challenge that could result from the gene therapy. Now, the ethical consideration for gene therapy. Now, the problem has to do with who decides which traits are normal and which constitutes a disability or disorder. Which traits? One may say that this is normal, the other may say that it is abnormal. Now, two, will the high cost of gene therapy make it available only to the wealthy or the rich? Now, after defining which trait is normal and which trait is not normal, even after identifying the one which is not normal, in a range, who will be able to, will be able to afford the cost of the therapy? Will it only be made available for those who are rich? Now, should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability, and the rest? That's also another concern or consideration, ethical consideration that will be raised. Obviously, people are going to abuse it if it happens like that. Now, two, the stem cell tech and therapeutic cloning. So let's look at the stem cell technology. The stem cells are a class of undifferentiated cells which are able to continuously divide and differentiate into specialized cell types. So in plant cell, we'll say the meristematic cells. So in human, we say stem cells because these are undifferentiated cells and they have the potency of differentiating or becoming more specific, growing to becoming more specific and specialized to forming other cell types. So stem cell divides. Each new cell has the potential either to remain stem cell or become another type of cell with a more specialized function, such as a muscle cell, a red blood cell, or a brain cell. Now let's look at the characteristics of a stem cell. They are blank cells, just like blank sheets. You haven't written anything on it. So they haven't differentiated. So in quotation, we say they are blank cells. Thus, they are unspecialized cells. They are also capable of dividing and renewing themselves for a long period of time. And number three, they have the potential to give rise to specialized type of cell. Okay, let's look at kinds of stem cells. Kinds of stem cells. We have totipotent cells, we have pluripotent cells, and we have multipotent cells. So totipotent cells. Now, each cell can develop into a new individual. For example, cells from early 1 to 3 days of embryos can develop into a new individual. Now, we have the pluripotent cells. They form any of the over 200 types of cells found in the human body. For example, some cells of the blastocytes, that is from between 5 days to 14 days. We have the multipotent cells, and they can only differentiate into a limited number of several types of cells. For example, the bone marrow contains multipotent cells that give rise to all the types of blood cells. We also have the third one. So first we look at the gene therapy. The second one we looked at the stem cell therapy by either using the pluripotent, the multipotent, or the totipotent cells. Now we hope to have the therapeutic cloning as well. So this involves extracting the nucleus of a cell and putting them into an egg, which is denucleated. Then allow it to divide and grow into a wide variety of different types of cells. The resulting cells, when used for treatment of the person, will have compatible genetic characteristics and immune reaction will be reduced. It is used to grow and replace organ, and also to create nerve cells for someone suffering from brain damage or a neurological condition. We also have the pharmaceuticals and the vaccines. Now let's talk about drugs. Pharmaceutical and vaccines, and then also the genes that encode clinically important proteins are isolated and introduced into bacteria. Bacteria that incorporate the recombinant gene synthesize large amounts of the protein the gene can specify. The protein are then extracted and purified. So for example, such products are insulin, which is essential for the control of blood sugar levels, and also the erythropoietin, which stimulates red blood cell production. We also have the second one, which is farming. So farmers use recombinant DMA. I'm not referring to FARMERS. I'm referring to the PHAMERS, the farmers. They use.
Please say what does it mean? Please explain this one. What does it mean?
Come again.
Please, the farmers. As initially I thought it was farmers, but he said farmers. Can you please highlight on it a little?
Yeah, so that's what I'm actually coming to, expose on. So what these farmers do is to use the recombinant DNA technology to create hybrid genes, okay, by crossing animals or plants DNA with genes that makes a desired protein. So we are not talking about the farmers that they apply inputs and what do you call it? They cultivate crops and all that. Rearing farm animals and all that. We are talking about those who work with the genes. Okay, those who work with genes. Recombinant gene. They are involved in the recombinant DNA technology, in essence. So the gene could come from human that they use, could come from human, it could come from bacteria, or it could come from other microorganisms. So they combine to form hybrid and then insert it into organism of host, or better still, in a host organism for them to express such characteristics. So in animals, the hybrid gene is introduced into embryos, whilst in plants, the hybrid gene is spliced into young plants. So farming has yielded drugs such as growth hormones and blood components such as the hemoglobin, but it is still in an experimental stage as manufacturing processes continues. We also have the DNA vaccination or DNA mediated immunization. So instead of giving you the protein version or the inactivated portion of the organism, that we, basically know. This time around, they are vaccinating with the DNA. So this involved direct introduction into host organism tissues of a plasmid. That's the plasmid DNA, which is able to cause expression of antigenic protein directly within the transfected cells. Now, this time around, just as I said, they are not, getting into you or putting into you a protein of a certain microbe for you to raise a certain antibody against it. No, or the inactive form of the organism being introduced into you for you to raise antibody against it. This time, the vaccination has to do with the introducing directly the gene into you by using the plasmid. I hope we have spoken, at length about plasmids, which is a vector, and we can introduce this directly or indirectly. South African scientists have used genetic modification to insert the vaccine for the cholera into bananas, so that when you take the banana indirectly, you are taking the vaccine for cholera. Incorporation of vaccine into fruits facilitates the vaccination of people in developing nations, where facilities for storage, distribution, and application of vaccine by injection or in oral form often do not exist. Right. Let's pause here. I'm going to have a water break for just only 5 minutes, and then I will come and then we continue with the xenotransplantation. All right. Hello.
Yes,
Hello
Doc.
there.
Hi.
All right. So I think yes, I think we can continue and move a bit faster. Okay, so let's look at the xenotransplantation. So xenotransplantation is all about the use of live cells and tissues or organs such as kidney, liver, and hearts from non-human animals species for transplantation into human patients. And we have had a lot of challenges with this type of transplantation. So technical possibilities of xenotransplantation have increased as knowledge of immune reaction has also grown. Just as I said, there has been a lot of immune reaction towards this xenotransplantation. The key target is to alter the cell surface recognition properties of the donor organs so that transplant is not rejected by the human immune system. So yes, they are still working around the clock to find solution to this type of transplantation. And I think it's better than using a fellow human, organ, like a kidney, letting somebody donate his or her kidney to you to go and do the transplantation. If that works for that of animals, I hope that goes a long way to help us in our existence as well. Because if one of yours is lost, there's no way you could regain it. And it is even difficult. And keeping a pressure on one isn't easy. So, ideally, if the two are all intact there, I think it's better. So, they are still working around it and finding solution to cell surface recognition, issues. If it is, it works out perfectly, then that's okay. I think if there is any improvement and it works out, that the human body does not reject some of these transplantation that occurs, then that's hallelujah. Goes a long way to help us all. So that is with the xenotransplantation. Let's look at the monoclonal antibodies. And the monoclonal antibodies are specific antibodies produced by clone from a single parent cell by fusing the antibody producing cell, that's the B, with a lab cultured myeloma cell through the process of somatic cell hybridization. So they hybridize the cell, and at the end of the day, you will have, composition of the two genes from the myeloma cell and then also the cell antibody in one cell. The result is a hybridoma. Now, the hybrid is called the hybridoma, which has the capability to produce monospecific antibody that can help in the recognition or purification of target cell. Now, due to their increased specificity, monoclonal antibodies have become powerful tool in medicine, and they are used for diagnosis purposes. For example, it includes the ELISA test for diagnosis of HIV and the AIDS. Now, we also have the targeted drug therapies, and I think this one will be much enjoyed. So the targeted drug therapy, they look at a specific gene sequence, which is, they produce drugs that is specific to, if it has to do with, I've forgotten your Emmanuel, they specifically produce a drug which is only going to work based on the gene sequence of that of Emmanuel, and then it will work perfectly. They are not going to do general drugs that will work for almost everybody. And these try and error stuffs that's happening in our hospitals and our health facilities will actually stop. So in this process, they use precise knowledge of the function of the gene and the protein or enzyme it produces. This enables scientists to produce a drug that is intended to act only on that particular enzyme or protein and produce a correction to its absence, deficiency, or even when it is in excess. Now, we also look at enzyme replacement therapy. Enzyme replacement therapy. Now, this involves, producing an enzyme that is administered to the individual or to the person as a medicine. So, in the case of insulin, they produce the enzyme and they inject the enzyme into the individual to correct the excess accumulation of sugar in the blood. So the enzyme then takes over the function of a similar enzyme that the person's own cells are unable to produce in the right quantity as a result of genetic default. Like I mentioned, if the person is diabetic, it means there's a lot of blood sugar, or the blood sugar level is that very high, and you don't have enough insulin to correct that, or better to coagulate and store it in the form of glycogen in the body. So they rather inject the patient with the enzyme so that the enzyme will work in replacement of the one that you are not able to produce. We'll look at our very last subtopic, which is bioremediation. Bioremediation. Please, up until this point, do we have any question as far as biotechnology, treatment is concerned? Any question? Then we go to bioremediation. Please, any question? We have about 30 minutes more. Please, any question? Are you with me?
Yes, please.
Yes, Doc.
Yes, sir.
Yes, Doc.
Okay. Right. So let's continue. So bioremediation.
Yes, sir.
What exactly bioremediation? What exactly are we talking about when we say bioremediation? I think somebody's hand is up. Bashiru, your hand is up. Bashiru, your hand is up.
Yeah, Doc. Please, sorry to drag you back a bit. I was having challenges in
No
my
problem
network.
at all. No
Please,
problem at all.
yeah, please, on this thing, the researcher, the Canadian researcher, who intended to grow tomato in
Kindly
a
speak up a
partial,
bit. Bashiru, kindly speak
yeah,
up a
please,
bit.
please, can you hear me?
Yes, I can hear you now.
Okay. I was saying that the
Yes, I can hear you
Canadian
now.
research, okay, the Canadian researcher who intend growing tomato on partial this is salty water or that is marine, and I wanted to know how will the tomato taste when tomato is grow in what marine water or partial salty water. How will it taste?
Yeah.
That's my question.
So what happens is that these are all biotechnological processes, okay? And what is going to happen is that he is going to actually look for a gene of a plant that is going to that is able to dwell in these habitats. Whether open lagoons or closed lagoons or swamps and all that. He's going to actually look for, I'm sure he has done that work. He's going to look for a gene which is salt tolerant, and is able to enable the plants grow in such a very salty environment. Now, there are some plants that are able to sequestrate salts in their system and exude them, or better still, excrete them, the excess salt from their system. Example is the Avicenna. Okay, the Avicenna plant is able to exude the excess salt that it absorbs. So yes, the scientists or the Canadian scientists that you made mention of can, go and extract this gene that is capable of, helping the plant, or better still, responsible for exuding this salt, excess salt absorbed by the plants out of the system of the organism, and insert it into the tomato so that it can grow in a very salty environment without any problem at all. Yes, we can also have a plant growing in a very drought, a very dry, when I say drought, a very dry environment, in an arid environment, even though it is not able, usually it's not able when it is not being engineered. We have some of them that have been engineered that even without raining for some period of time, they'll be able to withstand these droughts and all that. We also have some plants developed. I'm just giving you examples. We also have some plants being, developed or engineered so that they can grow in both acidic environments and then also alkaline environments. So yes, the characteristics are there. Yes, the characteristics are there. But when you are able to get the gene that is able to code for such characteristics, you can, extract it and, of course, inject the characteristics of beta cell, find a way of introducing that characteristics into another organism of interest. And that organism of interest will be able to exhibit that characteristics without necessarily changing the morphology, or better still, the composition of fruit or seed that it produces.
Okay.
Bashiru, I hope you are with me.
Yeah, I'm with you, Doc. Thank you very
Yes,
much.
yes. So it is possible. It is highly possible. The tomato fruit that will be produced will not be salty because that particular gene is going to act on exuding, or better still, taking off the salt from the organism. Just as I gave you an example.
Yeah. Thank you very much, Doc.
Yes, Julie.
Sir, please, I want to know how do these scientists they go by the extraction of those desirable traits?
How do they go about what?
The
I,
gene,
I
how do they go about those desirable traits that they pick from?
oh, what we have something called gene library. Okay, we have something called gene library. Now, with the gene library, we have almost all the traits. We have almost all the traits and characteristics of organisms in that gene library. We have all the traits and characteristics of organisms in that particular gene library. So, but still, they are still discovering some characteristics and traits, and those that has been, those that haven't been discovered, they are still sequencing their genes in that particular and stored in that particular library. Now, this is what happens if you find the organism and the specific traits that you, desire. You are going to extract the gene, and that gene is going to be sequenced and matched to that in the gene library. Okay, if it is able to match the sequence that you have, extracted, it is able. If it is able to match to that of the library, then it means that is the specific gene that you are looking for, and you can work with it. But if you extract that particular gene, and of course we have some gene variants, that is why we say allele. You can have alternative form of the same gene. But then, of course, generally we have sequences that match for a particular gene, like a gene that could for what Bashiru just said, drought tolerance, like salts or the saline environment that is able to withstand saline environment, highly saline environment, that is able to withstand pests, that is able to withstand weedicides, that is able to. So we have gene library that has all these sequences there. So you extract your gene and then you export your sample. If you're in Ghana, you export your sample and then they sequence it for you. And then they have a term, a technical term for it. They will just throw it in a gene library and sequence it. If it matches with that particular gene, then you are good to go. You can insect the gene into the host organism, and the host organism will start expressing that particular gene of desired traits for you. So that is how scientists they work, or the biotechnologies specifically. That's how they work.
All right, doc. So In inserting the
Okay.
gene, it is done under the microscope.
Right. Please, are we go to move on?
Hello, Doc. Hello.
Oh, I think we have done the direct method and the indirect method of inserting the desired gene. Yes, I think we have done that. The insertion of the desired gene. We have the both direct method and we have the indirect method. The direct method, we have the use of electroporation. We have the gene gun method and so forth and so on. We also have the indirect method where you use the bacteria. You insert the gene into a plasmid, place it in a bacteria, and you infect the plants with that particular bacteria. For example, is the Agrobacterium tumefaciens, which is causes the crown gall. I think we have studied that in our previous slides. Okay.
Thank you, sir.
Yeah. So moving on to bioremediation.
Hello, Doc.
Yes, sir.
My hand has been up. Please, with regard to these galamsey activities, can
Yes, sir.
biotechnology solve the chemicals that are released into the soil? Can we explore and get biotechnology solving this problem?
Yes, exactly. That's the topic that is on your screen right now. Bioremediation.
Oh, okay. Okay.
That's the topic which is on your screen right now. Bioremediation.
Hello,
Okay.
sir.
Did you hear me, please?
Yes, sir. Yes, sir. Please, sir, continue.
All right. So bioremediation actually is any process that uses microorganisms or their enzymes to return the environment altered by contaminants to their less toxic levels. Now, these levels must be below a limit which is established by regulatory authorities or to its original condition. So, just as uh, well, I've lost the one who asked the question, but we use microbes to correct instead of using chemicals to also, correct this harm that has been done to the environment. We rather use microbes. So, for example, oil spill on the ocean, we don't use chemicals to clean it. What we use to is that we introduce bacteria that is able to digest this oil, and then the whole place or the whole entire zone which had the oil spill is cleaned by these bacteria. Now, if it has to do with the use of plants to clean whatever harm that has been caused, whatever introduction of these heavy metals into the soil, we refer to that term as phytoremediation. We refer to that as phytoremediation. We will get to that. It is in a subsequent slide, so we will get to that. I don't want to jump the gun, so let's proceed accordingly. So the conventional method of remediating the environment includes soil, excavation, and landfilling, soil washing, immobilization or extracting by physiochemical techniques, high temperature incineration, and chemical decomposition such as UV treatments. So all these are treatments that are conventional. We have been using it over the years. The excavation, they excavate the site which has been contaminated. We also landfill, we do landfilling. There's something that we have been doing, and we also do soil washing and immobilizing or containing the area that has been, contaminated. We also do the physio or physical chemical techniques and then high temperature incineration and all. They have not been helping. At the long run, they come back and they hunt us in a way that we are not able to, reverse them. So what happens is that we have to resort to reducing, microorganisms. At the end of the day, they will be able to, if not to a tolerable level, they will be able to reduce it to a very low limit to which it cannot be, dangerous to our health. So let's look at the remediation in detail. So let's look at factors affecting bioremediation. The existence of microbial population capable of degrading the pollutant is a challenge. Microbes. If the pollutants are much, it will be just quite difficult for these microbes for them to digest, or better still, for them to bring them to normal, less tolerable, or better, less harmful levels. So the availability of contaminants to the microbial population, the availability of contaminant to the microbial population, sometimes it seeps down and leaks deep down in the soil, that it becomes quite difficult for the microbes to act on them. Then the environment of factors, or the environmental factors such as type of soil, temperature, the pH, the presence of oxygen or other electron acceptors and nutrients suitable for the microbial population to act on these contaminants is also a serious issue under consideration. Now let's look at classification of bioremediation techniques. Classification of bioremediation techniques. We can do on-site or outside the site, remediation. If it is on-site, then we are looking at in situ. In situ. In situ. That's the on-site. And then if you are moving the contamination from away from the site, we say we are doing ex situ remediation. So let's look at the in situ. So in situ involve treating the contaminants material at the site to avoid excavation and transportation of contaminants. It is the most desirable and it has less disturbance, less cost, and is limited by the depth of soil that can be treated. All right, then we have the ex situ, which involve moving the contaminant out of sight. So this involves the removing or the removal of contaminated material to be treated elsewhere. Some example of in situ treatments include in situ, that's the on-site treatment. We have bioventing, that is supplying air and nutrient through the wells to contaminated soil to stimulate the indigenous bacteria to digest, or better to degrade this pollutant. Then the in situ biodegradation. This involves supplying oxygen and nutrients by circulating aqueous solution. So one is, supplying air, and this one has to be the degradation. In situ degradation has to do with supplying or circulating the aqueous solution through contaminated soils to stimulate naturally occurring bacteria to degrade organic contaminants. So the venting has to do with passing, supplying accurate, or right amount of oxygen for their respiratory activities and as they degrade. All right. Let's look at, let's still continue and look at the in situ form of, remediation. The next one is biosparging. The injection, this has to do with injection of air under pressure. Injection of air under pressure below the water table to increase groundwater oxygen concentrations to enhance degradation of contaminants by bacteria. And then bioaugmentation is also in situ technique. And this has to do with introduction of natural microbial strains or genetically engineered variants to contaminant or the contaminated sites so that degradation will be achieved. So you look at the specific contaminants and you introduce the natural microbial strain that will be able to, work on that pollutant or the contaminant. That is, you are augmenting it. Now, biostimulation. This is the modification of the environment by addition of various forms of nutrients. That is nitrogen, oxygen, and other, very important gases to stimulate growth of bacteria for degrading the pollutants. Now, we look at the ex situ treatments. The ex situ treatments. And the ex situ treatment, we have land farming, we have biopiles, and we have the bioreactor. These are all the ex situ treatments. Now, land farming, we have it as contaminated soil is excavated. We have it ex situ, so we are moving out of sight. We are moving the contaminant out of sight. So the land farming, what you do is that you move the contaminated, soil, and then you go and spread it over a prepared bed and periodically tilled until pollutants are degraded. Then we have the biopiles. Then with this, the excavated soil are mixed with soil amendments and then formed into compost piles and then enclosed for treatment. Now we have the bioreactor, which has to do with processing of
Hello,
contaminated. Yes, sir.
Please, sorry. With the soil amendments,
Okay.
what substrate do you use to amend the soil?
Oh, we
Is
can
it
use.
is it organic ones or what?
Yes. Yes. So you mix it with the compost, like the compost file. Compost. The compost, the animal dung and all that. Okay, you mix it with the what do you call it? The pollutants. You mix the soil which has been contaminated, okay, with this cow dung and the other. There are some bacteria that exist in digesting this cow dung and those kind of things into compost, which are very beneficial to plants nutrients and then absorption. So
yes, sir,
you mix the contaminated soil with this the amendments that we are mentioning, okay, the compost, okay, and then you allow the bacteria to act on it. But most of the times these contaminant that we are referring to, we have specific bacteria that act on them. We have specific bacteria that on them. So we have specialists that are able to detect those contaminants that are found in specific areas, and the bacteria that is able, or the microorganisms that are able to degrade these pollutants. It's not every bacteria or it's not every microorganism that will be able to act on specific pollutants. Please, are you getting what I'm saying? But
very
just to
well, sir.
facilitate the process of decomposition of these contaminants, we have to amend it, or better, we have to augment the soil with these compost piles. Are you getting what I'm saying?
Yes, sir.
So you have to just add these compost to facilitate the process of decomposition of these contaminants. Right. So the bioreactor is the processing of the contaminated solid material, that is soil sediment or sludge, or water through an engineered contained system. Through the engineered containment system. So this one, you don't spread it. What you have to do, you have a containment. It's a machine. The bioreactor is a facility, or better still, is a specific machine that is responsible for decontamination, or better still, for reducing the toxic levels of the soil. They feed the contaminated soil into the bioreactor, and it is able to reduce its toxic level by absorbing some harmful minerals or some harmful contaminants from the soil, and then afterwards the soil is then redeposited at either their original sites, or they are dumped into areas that are going to be used or reused. Now let's talk about the advantages of bioremediation, and then we'll talk about some disadvantages. Then we go to phytoremediation, even though it's left with just a minute of our time to hit 8 p.m. But I'm hopeful that we will be able to finish up with this. Yeah. Right. So, advantages. We have it as it being less expensive. It's, causes less disturbance or disruption of human activities. And we have possibility of complete destruction of pollutants. It is perceived to be an acceptable one, and because it poses no risk, it's perceived to be the acceptable form of degradation of pollutants, and it poses no serious health or safety risk to employees involved in the degradation, because at the end of the day, you're not going to burn them, okay, so that the fumes will be inhaled. So it can remediate areas that are not easily accessible or inaccessible to other technologies. But let's look at some disadvantages as well. So these are concerns. These are concerns that, or there are concerns. I'm sure that should be the caption. I'm supposed to make it that. So correct that for me, even in your reading. So there are concerns that products of biodegradation may be more persistent or toxic than the parent compound. So that is a concern. That's one concern that has been raised. And also limited to biodegradable compounds. So if the compound is not actually going to be, or is not going to be let me put it technically that if the compound is not going to be able to be degraded by the living organism, it will be quite difficult. It will be quite difficult for the such remediation to occur. Piedu, I don't know whether you have something to say because your mic is on. Nyako and Piedu. Right. So essentially what we are saying is that the second point is that there are some compounds that are not going to be degraded. Example, the heavy metals. It will be quite difficult for some microbes or other microbes to act on them and degrade them. But then there are other organic compounds that are going to, that will be easily degraded by the organisms. So often takes longer than other treatment options that we have mentioned, whether incineration or the, land farming and the rest. So it is actually difficult to extrapolate from lab bench and pilot scale studies to full scale field operations. Yes, you can do some extrapolations at the lab, but carrying out a full scale field operation is quite difficult. It's not easy like that. And then also evaluating performance of bioremediation is difficult. There is no accepted definition of clean, and I like this portion, that there is no acceptable global definition of clean. What might be clean to you may not be clean to somebody, even though you might do so well in enforcing that the place is, completely degraded. But of course, there might be some residues left by these microorganisms. On that note, we go to the phyto remediation. We have about 1, 2, 3, 4, 5, 6, 7. We have about 7 slides to end our session. Do you make it next week? Will you make it next week?
Yes, Doc.
And add it to the unit.
Yes, sir.
Yes, sir.
Yes, Doc. Yes,
I'm sure unit 4 we can use some 1.5 hour to finish up unit 4. So can we add the phytoremediation to unit 4 next week? Are you okay?
Yes, please.
Yes, please.
Yes, of it.
Yeah, okay.
So do we add the
Yes, please.
phytoremediation to
Yes, that will be
unit 4?
fine.
Unit 4 is not that much.
Yes,
So if we can add the
please.
phytoremediation part
Yes,
to,
please.
okay, all right. So you can also read around the phytoremediation, and we meet next week. That is Saturday, and then we complete the unit 4. Please, within the week, I will send you. Within the week, I will send you your groups for the presentation of units 5 and 6, because we are almost done with the units that I was supposed to cover from unit 1 to 4. So next week, hopefully, if God gives us life, we will be able to finish up with unit 4, which means that it's left with your portion of unit 5 and unit 6 for you to complete the course. So I'll send you I've had your details. So I'll send you, irrespective of where you are, you can be in what do you call it? Temali.
Any
You can be in Temali. You can be in Temali and your presentation partner will be in second D. You can be in Tamale and your presentation partner will be in second D. So please take note and you act accordingly. Right. Frank, your hand is up.
Yeah, Doc, I think next week is our last week in the work. The following week will be for practicals, and then the preceding week will be quiz, and then exams follows.
This time there's no practicals.
I,
It's
don't- I
for
don't get you.
physics.
I don't get you, please.
Hello, sir.
I'm saying that
Let me help my friend.
this week is the last week for our meeting.
Come again.
I'm saying that we're supposed to have 4 meetings, so next week we'll end the 4 meetings. All things being
Yes,
equal.
that is exactly what I said. Or
But we said we still have some presentations to do. So when are we going to do that one?
you're supposed to do the units 5 and 6. You're supposed to do the units 5 and 6 as a
Yes,
presentation.
yes, Doc, I understand, though, but
Mhm.
the next week will be our last meeting with you.
Eh,
Sir, can we meet on the Wednesday again? Hello, sir.
Wednesday is for PCK.
No. It's not Wednesday.
It's Thursday.
No, PCK is Thursday. Then we met him this week, Wednesday. So I'm
Thursday is for PCK.
At
I don't
times.
know. Has
Hello.
she added the
Hello.
Wednesday?
Hello.
Please, but we were
Hello.
suggesting that day.
Hello.
Hello, sir.
PCK is on
Yes.
Tuesday. So if we did it on Thursday, it doesn't mean that it is Thursday. She didn't get time on Tuesday. And remember, she has given us somebody who's going to take charge of the class again. Yeah, that is why Nana Echo, what the name is there. Madam Abigail says her time is so tight. So after tomorrow, we go back to the original schedule.
Okay.
You get it. We go back to original schedule. So
You mean
some
the
of these
Tuesday?
things
So does that mean that we will not meet on Thursday for PCK?
Please,
Yes.
the recording is
Hello?
on. If there's anything, let's end the class, then we'll do the discussion, please. My humble opinion,
You have to be,
because the recording is still on.
you have to be settled. I thought they'd be now. By the time you realize they finish the lecture behind you, you have to be settled on the date.
Off.
Can we leave?
It's my doctor is off.
That's what I'm.
Hello, sir.
Yes.
Sorry, my this thing went off.
It's normal.
So yes, please, is there anything that is different from what I said? I need to know. I know next week is our last meeting as in me teaching, and then also the next week is for practical. The next week after next week is practical. That is next 2 weeks is practical. If I should put it right.
Okay, next week is practical.
Is that not true?
Yes, sir.
So we have to find a way of. Please, I'm listening.
Maybe we can meet within the week, one of the days. In the evening.
For the presentation. For the presentation.
Presentation or the continuation of our lectures.
Oh, no. For the lectures, the next week, God willing, when we meet, we'll finish up.
Okay.
And for the lectures, the next week, when we meet, we'll finish up for units 1, 2, 3, and 4. We'll be done with God willing.
Then maybe say the presentation can do that.
Yes. So maybe within the weeks, I would have to schedule and then give you some days for you to do the presentation. Okay.
All right, sir.
All right. Okay. All right. Do have a good evening. And whatever happens, I'll communicate it to your course rep as well.
Oh, okay, sir.
All right.
Thank you, sir.
You're welcome.
Thank you, sir. Thank you.
Bye. Have a good evening.
Have a good evening, sir.
Thank you. Bye.
Okay. Bye.